Hello everyone, welcome back to my channel IGCSE Biology Help. In today's video, I'm going to be doing the full syllabus explanation for the Cambridge IGCSE Biology 0610 or the 0970. This is the same subject, but the 0970 is for the 9 to 1 grading system. So I'm going to be explaining everything you need to know, and you can use this video as a revision before your exam. This is everything you need to know about the syllabus or the information that is coming for you in your exam. Make sure to subscribe and like so you can get more content like this. Let's start explaining. So first of all, we all know that our papers, they either come in paper 2, 4, or 6. This is actually for the extended. So paper 1 is for the core and paper 3 is for the core. But for the extended, you know that you're going to get a paper 2 multiple choice, and paper 4, this is for the extended. It's the same theory paper, but for the core they have it as paper 3, which is a bit easier than the extended. And of course, the practical test, some people take it and some people will take the alternative to practical. The alternative to practical, both extended and core students take it. So this is just an overall summary of the assessments. So let's start actually explaining. Okay, now chapter 1, this is the first thing that you're going to learn if you're taking IGCSE biology. So number 1 is the definitions. This is over here, if it's written core and there's nothing in the supplement is for the extended students. There is no extra information you need to know if you're an extended student. So everyone should know that the movement is an action done by an organism or part of an organism causing a change of position or place. This is the definition of movement. Respiration is the chemical reactions in cell that break down nutrient molecules and release energy for metabolism. This is Mrs. Gran. If you guys watched my previous videos, I've always, when I was solving questions, I told you that Mrs. Gran, it stands for movement, and R is respiration, and S is actually sensitivity, G is growth, R is reproduction, E is excretion, and N is nutrition. So this is just an easy thing you can remember to remember the living organisms like characteristics. So if anything is living, then it should have some or all the traits that I have just explained. Now, sensitivity is the ability to detect or respond to changes in the external or external environment. So growth is a permanent increase in size and dry mass, and reproduction is the process that makes more of the same kind of organism, and excretion, it's the removal of waste products of metabolism and substances in excess of requirements. G is the last one, which is nutrition, as taking in of materials for energy growth and development. So this is all memorization. You have to memorize all of these because they're going to ask for either one of them or some of them will come in your multiple choice. So you have to know all of these memorization. Now for 1.2, over here, there is extra information for the extended students, and this is just for the core students. So let's first see what the core students have to learn. They have to know that organisms can be classified into groups by the features that they share. So this is just an information for you to know. And they have to describe species. This is a definition you have to memorize. Species are a group of organisms that can reproduce to produce fertile offspring. And also, you have to describe the binomial system of naming species as an internationally agreed system in which the scientific name of an organism is made up of two parts showing the genus and species. So, this is also memorization. This is like a definition. So, you have to know all of this stuff. And I recommend that if you study from a teacher's notes or if you study from the book, after you finish studying, you have to print out the syllabus and you have to read out read out everything in the syllabus for this chapter that you just finished, and see if you understood everything that is written here, because this is very important that you know all of these stuff. Now, for the fourth part, it says construct and use dichotomous keys based on identifiable features. So, dichotomous key is actually in paper 4 only. It is a question where they tell you go to number 4, go to number 3. I've solved questions like these in my paper 4 solving videos. You can check them out if you want. So over here in the supplement, which is the extended students have to know more information, which is explain that classification systems aim to reflect evolutionary relationships. So classification systems, they actually teach us about relationships of animals. So for example, if we share genes with other organisms, and it also shows evolution of like the human species and like evolution of animals. So this is what you need to know. And also number 6 says explain that the sequence of bases in DNA are used as means of classification. So this is over here, if you're a core student, you only study from this part. If you're an extended student, then you study from the core part and you also study from this area. So if you're a core, don't study from this area. You just study from the area that is written on it, core. Now, also, you have to know number 7, explain that groups of organisms which share a more recent ancestor are more closely related and have base sequences in DNA that are more similar than those that share only a distant ancestor. Now, for 1.3, the next part of chapter 1, core students have to know that they have to state the main features used in to place animals and plants into appropriate kingdoms. So you have to know how to place an animal or a plant or anything into kingdoms. You have to know that. And also you have to state the main features used to place organisms into groups within the animal kingdom, limited to the main groups of vertebrates, mammals, birds, reptiles, and amphibians, and fish. So you have to know that there are in the animal kingdom, there are vertebrates. Vertebrates are animals that have backbone. They are mammals. We are mammals, humans, and also some other animals. Birds, such as a parrot or a chicken. Reptiles, and amphibians and fish. And also there is arthropods, which means animals that don't have a backbone, such as myriapods, insects, arachnids, and crustaceans. So you have to know all of these animals, and you have to know the groups and some examples for each of the groups of vertebrates or arthropods. And also you have to classify organisms using the features identified in 1.1, 1.31, and 1.32. So over here for the extended students, they have to know that they have to state the main features used to place all organisms into one or of the 5 kingdoms: animal, plant, fungus, prokaryote, and prototist. So over here in number 5, you have to know that there is also in the plants, there is ferns and flowering plants, dicotyledons and monocotyledons. So you have to know that there is dicotyledon, which means a plant that has 2 seeds, and monocotyledon is a plant that has 1 seed only. Now you have to also classify organisms using the features identified in 4 and 135. And you also have to know state the features of viruses limited to a protein coat and genetic material. So you have to know that a virus is made up of protein coat and genetic material for the extended students. Now let's move on to chapter 2 of the IGCSE Biology syllabus. We have 21 chapters. So chapter 2 is the cell structure, and in this section in 2.1, all this information is to be known by the core students and the extended. There's no extra information for the extended students. So we have to describe and compare the structure of a plant cell within an animal cell with an animal cell, limited to cell wall, cell membrane, nucleus, cytoplasm, chloroplasm, ribosomes, and mitochondria, vacuoles. So you have to know that plants and animal cells, they have organelles. Organelles are cell wall, cell membrane, nucleus, and everything that is written down over here. You have to know all of them, and you have to know that they even exist. So let's move on to number 2. Describe the structure of a bacterial cell, limited to cell wall, cell membrane, cytoplasm, ribosomes, circular DNA, and plasmids. So you have to know that a bacterial cell has extra stuff other than the animal cells. They have cell wall, they have cell membrane like us, they have cytoplasm, ribosomes, but they have circular DNA and plasmids. This is the extra thing in bacterial cells. So also you have to know that the functions and the structures listed in 2.11 and 2.12 in plant, animal, and bacterial cells. So you have to also know the functions of every single thing that is listed over here, like the cell wall. What's the function of the cell wall? For example, it is a fully permeable like cell that allows all structures to go through. And you have to know, for example, the nucleus is used for protein making, and you have to know all of this stuff, so you have to memorize that as well. And you have to also know that the new cells are produced by division of existing cells, and you have to know that specialized cells have specific functions limited to ciliated cells. You have to know the functions, and you have to know the specialized cells. So the first one that you have to know is ciliated cells, the movement of mucus in the trachea and bronchi. So there is ciliated cells in the trachea and bronchi, and you use them. It is used for moving the mucus. So it is used for moving the mucus around your throat and like that. So root hair cells, they are actually in the plants. They are used for absorption of water and ions from the roots, so you have to know that as well. And palisade mesophyll cells, these are actually in the plant. They are used for photosynthesis. And also the neurons in our nervous system, they are used for conduction of electrical impulses. And the red blood cells, they are used for transport of oxygen. Now, the sperm and egg cells, the gametes, these are used for sexual reproduction. They are used for, of course, reproduction over here. So, number 7, it says describe the meaning of terms cell, tissue, organ system, and organisms as illustrated by examples given in the syllabus. So, you have to know the definition of cell, tissue, organ system, and organism. These are all memorization. Now, they didn't write down like the definitions, but you can find them if you are studying from the book or a teacher's notes. Now, 2.2, the size of specimens. This is the I've explained this in my paper 6 video if you want to watch it. This is basically the I am triangle that I've explained before. This is used to calculate actual size of an image, or the magnification, or the image size. So this is very important both for core students and extended students, because you're going to use this in paper 6. So, as I told you in the beginning of the video, paper 6 is taken by both core and extended students. So you have to know this stuff. You have to know that the magnification is calculated by image size over the actual size. This is basically what is shown over here from the graph, or from the triangle. So if you want to calculate magnification, you're going to do image size, which is I, and actual size over the image size. So if you want to calculate magnification, you're going to be using millimeters as units, or you can also use micrometers if you are an extended student. If you want to convert from millimeters to micrometers, you have to multiply by 1000. So for example, if you have 10 millimeters and you want to convert it to micrometers is less than millimeter. 1 millimeter is equal to 1000 micrometers. So you're going to multiply 10 by 10,000, 10 by 1000, and this is going to give you 10,000 micrometers. This is just an example. You can also know more again in my paper 6 video. Now this is the next chapter, chapter 3, movement into and out of the cells. This is a very important chapter. It comes in all papers. So you have to study this chapter very well, because I see it all the time in the past papers. So over here in this section, you're going to have to know that diffusion is the net movement of particles from a region of their higher concentration to a region of their lower concentration down a concentration gradient as a result of their random movement. This is memorization. You have to memorize this definition. This is important. And also, number two says that the energy for diffusion comes from the kinetic energy of random movement of molecules and ions. So diffusion and actually osmosis, they don't need energy like the active transport. They don't need respiration. They just get their energy from the kinetic energy of random movement of molecules and ions. So this is non-passive. 3 says state that some substances move into and out of the cells by diffusion through the cell membrane. You have to know that stuff moves out and into the cells by diffusion. Diffusion is how, for example, if you spray a perfume at the end of the room, you can smell it on the other side of the room. By diffusion, the air molecules will move and they will diffuse to the other areas. So this is just an idea of diffusion. Now describe the importance of diffusion of gases and solutes in the living organisms. You have to know that, and you have to also know investigation the factors that influence diffusion limited to surface area, temperature, concentration gradient, and distance. What does that mean? This means that you have to know that diffusion is affected by several factors such as surface area. If there is larger surface area, the diffusion will be slower, and if there is higher temperature, then there is a higher rate of diffusion. And concentration gradient, if there is a high concentration gradient, there is more diffusion. And distance, if there is a high distance, there is a slower diffusion. Let's move on to osmosis, which is the movement of water. Now we have to know in this area there is extra information for the extended students. Now describe the role of water as a solvent in organisms with reference to digestion, excretion, and transport. So you have to know what is the role of water. Memorize this, that water is a solvent, and also state that water diffuses through partially permeable membranes by osmosis. This is again memorization. You have to also understand this concept very well. 3 says state that water moves into and out of the cells by osmosis through the cell membrane. You have to investigate osmosis using materials such as dialysis tubing. So dialysis tubing, you have to understand the concept of dialysis tubing. This is something that is used to show an example of osmosis and how water can perform osmosis and how some stuff can diffuse. And number 5 says investigate and describe the effects on plant tissues of immersing them in solutions of different concentrations. So this means, for example, like if you put a plant in a very concentrated solution, then it will actually shrink because all of the water will move from high to low water potential. So this will cause the cell to shrink. If you put it in a very high concentration of water, if there is more water outside, then the plant will actually grow in size and it might actually like burst. So you have to know all of this information. And also number 6 says state that plants are supported by the pressure of water inside the cells pressing outwards on the cell wall. So now for the extended students, they have to describe osmosis as the net movement of water molecules from a region of higher water potential to a region of lower water potential concentrated solution through a partially permeable membrane. So you have to know this definition by heart. This is very important, guys. You have to memorize this. And this is how you describe osmosis in your exam. Don't forget to write higher water potential, not higher concentration. Higher concentration is wrong when you are describing osmosis. Make sure to write from a region of higher water potential. Now, number 8 says explain the effects on plant cells of immersing them in solutions of different concentrations by using the terms turgid pressure, plasmolysis, and flaccid. So turgid, it actually means a plant that is like very hard because there's a lot of water inside, and flaccid means like a plant that is like I don't know, it's like going or bending downwards. So this is flaccid. Plasmolysis is like the movement of water. So you have to know these terms and you have to understand what they mean. Now number 9, it says explain the importance of water potential and osmosis in the uptake and loss of water by organisms. So you have to know this as well. You have to know that the water potential and osmosis, they are used in the uptake and loss of water by organisms. So this is how the organisms take their water. And you have to also know that water potential is very important in the life of a plant. So if there is not a right water potential, then there is a problem that can happen with the plant. Now, for active transport, this is the process that uses respiration to release its energy. So, for the core students, you have to describe active transport as the movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration against a concentration gradient. This is very important. Sometimes in the exams, you can take just 1 mark if you say this. And using energy from respiration, as I just said, this is a process that needs energy and respiration to occur. So this is all that core students need to know. Now, for the supplement or the extended students, you have to know the importance of active transport as a process for movement of molecules or ions across membranes, including ion uptake by root hair cells. Now, active transport is very important for plants because it helps the plants to take the ions and water from the root. When you give water to a plant in the soil, then it will use active transport to take the water and ions from the soil, from the roots. So this is very important again. Now, number 3, you have to know that protein carriers move molecules or ions across a membrane during active transport. So there's protein carriers in the cell membrane. So if, for example, there's a cell that has a cell membrane, there will be something like this. It is called a protein carrier. It will open when there is active transport, and it will close if there is no active transport. It will allow, for example, this is a particle that wants to enter. It will open like this, it will let it inside, and then it will close again. So this is very important. You have to understand that. Number 4, which is the chapter 4, biological molecules. So you have to know, for the core students, you have to list the chemical elements that make up carbohydrates, fats, and proteins. So carbohydrates are made of carbon, they are made of hydrogen, they are also made of oxygen. Now, also, this is the same thing for fats. And proteins, they are made of carbon, hydrogen, and oxygen, but you have to add nitrogen as well. This is only for the proteins. All the others, carbohydrates and fats, they are only made up of carbon, hydrogen, and oxygen, except for the protein that has extra nitrogen. Now, you also have to know that you have to state that large molecules are made from smaller molecules limited to starch, glycogen, cellulose from glucose. So we have to know that glucose, actually, it makes up 3 things. It makes up starch. Starch is used as like a sugar storage. So, for example, if there's no energy in the plant, then it will break down the starch and release energy for itself. And also, glycogen, it is in humans. It is stored in the liver and the muscles. The glycogen, the body uses the glycogen to break down the sugars if there's no energy. And cellulose is in the plant cell. It is used for making cell walls. So this is all made from glucose. Now, amino acids, they make proteins. So proteins are made from amino acids, and fats and oils, they are made from fatty acids and glycerol. So actually, you have to know that there is 3 fatty acids and 1 glycerol. This is the exact information that you have to know, because sometimes they will ask for it. So 3 fatty acids and 1 glycerol. So you have to also know the use of iodine solution test for starch. Now, if you want to test for starch, you're going to use something called the iodine solution test, and you also need to know the colors. I have explained this in my paper 6 tips and tricks videos, and you have to know the Benedict's solution test for reducing sugars. So if you want to test for reducing sugars, you have to know the Benedict's is used for it. And also, reducing sugars is all sugars except for sucrose. So if you want to test for sucrose, then you can't use the Benedict solution because it is non-reducing. Biuret test, it is used for protein testing. And if you want to test for fats and oils, you can use ethanol emulsion test. And vitamin C is used, if you want to test for vitamin C, you have to use the DCPIP test. You also have to know the colors. If there is protein, then the color will change from blue to purple, for example. You have to know all of these colors. Now, number four, this is actually only for the extended students. So if you're a core student, you don't have to know this part. It says you have to know the structure of a DNA molecule. Actually, this is a memorization question. It sometimes just comes in the exam. It just says describe the structure of a DNA molecule. You have to know that the DNA is made up of two strands coiled together to form a double helix, and each strand contains chemicals called bases. And the bonds between pairs and base of the bases hold the strands together. The bases always pair up in the same way, A with T and C with G. Full names are not required. You don't have to know these. You will know the full names if you take advanced level. Now you have to know that a base that is A, it has to pair with T. And if it's C, you have to bind it with G. If it is the opposite or if it's anything other than that, then it is wrong. Enzymes, this is chapter 5. You have to know for the core students, you have to describe a catalyst as a substance that increases the rate of chemical reaction and it is not changed by the reaction. Now, what does that mean? It means that an enzyme, a catalyst, is an enzyme and it is not changed by the reaction. It means that it is not used up. It is something that stays in the same quantity and it's not changed. So 2 says describe enzymes as proteins that are involved in all metabolic reactions where they function as biological catalysts. That means that enzymes do the same job as catalysts, but they are actually made up of protein and they are in all living organisms. Describe why enzymes are important in all living organisms in terms of a reaction rate necessary to sustain life. So, enzymes, they are made so that you can have, for example, if you eat a piece of meat, then you can digest it in like 4 hours or 3 hours or sometimes 2 hours. If you don't have enzymes, then it will take you maybe maybe a year, to digest this piece of meat. But enzymes, they make this reaction time much faster so that you can sustain life. Now 4, it says describe enzyme action with reference to the shape of the active site of an enzyme being complementary to its substrate and the formation of products. So you have to know how the enzyme action works. So you also have to talk about the active site shape. So for example, if you have an enzyme, enzymes work by binding to the substrate shape. So for example, if you have a piece of fat, there is, for example, the enzyme for fat, it will look like this. So this piece, it will actually bind over here. This area, this thing that it binds to, this is actually the enzyme. This is the enzyme, and this piece, the circle, is actually the substrate. This is the thing that is being broken down. So you have to know all of this, and you have to know that the enzyme has to be in complementary shape, which means the shape of this substrate, it has to be the same as and it has to fit into the enzyme in a complementary manner. Investigate and describe the effect of changes in temperature and pH on enzyme activity with reference to optimum temperature and denaturation. So you have to know that enzyme, it is affected by things such as temperature. So if there's extremely high temperatures, the enzymes will not work well and they will slow down the reaction rates, and this is a big problem. And also the pH. If, for example, some pHs, they will only work in, for example, let's say pH 7. If the pH is changed, if it is decreased or increased, the enzyme will not work as it should. Optimum temperature and denaturation, this is exactly what I'm saying. Optimum temperature is that perfect temperature where the enzyme can work. And denaturation is when the enzyme shape will change. So for example, it is supposed to look like that. But after denaturation, it will basically melt down and the shape maybe will change into something like this. So if, for example, there's this substrate, it will try to bind over here. It will not work because the active site, this thing, changed shape. So it will not work anymore. So this is what is denaturation. You have to know all of this, of course. Now, for the extended students, there is extra information. They have to explain enzyme action with reference to active site, enzyme, substrate, complex, substrate, and product. So this is basically what I just explained. And there's like specific keywords that you have to know, such as product. Product is the end or the thing that comes out after enzyme is broken down. So, for example, if I break down if I break down a piece of starch, then it will bring out glucose. This is what happens after it's broken down. This is the product, basically. Number 7, it says to explain the specificity of enzymes in terms of complementary shape and the fit of active site with the substrate. So we have to say that enzymes, they have specific shapes and complementary shapes that have to bind in order to create an active site substrate. And active site substrate, it is crucial so that the enzyme can work and break down and actually do its job, basically. Number 8 says explain the effect of changes in temperature on enzyme activity in terms of kinetic energy, shape and fit, frequency of effective collisions, and denaturation. So this means that if there is a change in temperature, there is a change on the enzyme activity, it may slow down or it may increase. And also the frequency of effective collisions, which means that if there's high kinetic energy, then there is higher enzyme activity, and denaturation, of course, it will make the enzyme not work at all. Now, the last thing you have to know is that you have to explain the effect of changes in pH on enzyme activity in terms of shape and fit and denaturation. So basically, you have to know what happens if the pH changes and how it will affect the enzyme activity, basically. Now, for chapter 6, which is plant nutrition, you have to know that 6.1 is about photosynthesis. This is basically the basic thing that a plant uses to create its own food, and it uses photosynthesis. So the plant doesn't actually eat and drink like humans. It actually does something called photosynthesis, and this is basically a process by which plants synthesize carbohydrates from raw materials using energy from sunlight. So this word synthesize, this actually means make. So plants make glucose, they make sugar from raw materials using energy from light. So photosynthesis actually works by the plant, it will absorb carbon dioxide and it will absorb water, and it will use enzymes, and it will create glucose and oxygen just from carbon dioxide and water. So this is the basic thing that is needed for plant life. So chlorophyll is the green pigment that is found in chloroplast. So basically, chlorophyll is the whole mastermind of everything in the photosynthesis. It will create the glucose and from the carbon dioxide and water. So this is very important. Chlorophyll is only found in plants, so be careful. And it's also green pigment. So this is what makes the plant green, the leaf green. Now you have to know that chlorophyll transfers energy from light into energy in chemicals for the synthesis of carbohydrates. So chlorophyll, you have to know that it will transfer energy from light into energy in chemicals. It will basically transform light energy, which means the sunlight from the sun, light energy into chemical energy, which is the glucose, into chemical energy. So you have to know that, and this is basically the job of the chlorophyll. Now, for the extended students, they have to know the balanced chemical equation for photosynthesis. If you studied chemistry, then you already know that this is the balanced equation. Basically, 6 carbon dioxides and 6 waters. This is the raw materials for photosynthesis. It will create glucose, which is 6 C6H12O6, and 6 oxygen molecules. And now this is the continued part. Now you have to know, for core students you have to know, and also for extended students you have to know the use and storage of carbohydrates made in photosynthesis limited to starch as an energy store. So basically you have to know what starch is used for. I have explained this in like chapter 2. I've told you that starch is used as an energy store in plants. Cellulose is used to build cell walls. Glucose is used in respiration to provide energy. Um, so this is basically the thing that makes the plant live. Glucose, it uses it in chemical equations, I mean in chemical reactions, so that it can create energy and live. Sucrose for transport in the phloem. Nectar to attract insects for pollination. I will explain phloem later on in the later part of this chapter. Nectar is to attract insects for pollination. Explain the importance of nitrate ions for making amino acids and magnesium ions for making chlorophyll. Now, uh, the nitrate ions are making amino acids. Amino acids, as I explained before, it is used for protein making. Protein making actually will help the plant to make like DNA. It will help the plant to make cell walls. It will help the plant to make stuff from protein. So this is very important that the plant has nitrite. Now magnesium ions are for making chlorophyll. If there is no magnesium in the plant, it will turn yellow and therefore it will not be effective in making photosynthesis. Number 7 says, investigate the need for chlorophyll, light, and carbon dioxide for photosynthesis using appropriate controls. So you have to basically know that the plant needs chlorophyll, light, and carbon dioxide for making its food by photosynthesis. Number 8 says, investigate and describe the effects of varying light intensity, carbon dioxide concentrations, and temperature on the rate of photosynthesis. So this means that if you change the light intensity, for example, if you have high light intensity for a plant doing photosynthesis, then the photosynthesis will actually increase, like it will be faster. But if there's low light intensity, the light is not as strong, then the process will be a lot slower. Also, the carbon dioxide concentration, if it increases, then the photosynthesis will increase because there's a higher amount of raw materials available. And if you decrease it, of course the process will be slower. The same thing applies to temperature. This is because the enzymes, if you really increase the temperature, then the enzymes will be denatured. If you decrease it, then the enzymes will not be working in their optimum temperature, as I explained in chapter 5. Number 9 says, investigate and describe the effect of light and dark conditions on gas exchange in an aquatic plant using hydrogen carbonate indicator solution. So if there is dark conditions, then there will be no photosynthesis, of course, because there is no light, and we know that it requires light and carbon dioxide. So there will be no light, so it will respire. It will not make any photosynthesis. It will respire and it will break down the starch so that it can release energy for itself. So this is what happens to the plant if it's in a dark solution. And you also have to know the colors of the hydrogen carbonate indicator. So also for the extended students, there is an extra information. It says, identify and explain the limiting factors of photosynthesis in different environmental conditions. So limiting factors are stuff that may prevent the photosynthesis from being at its maximum performance. So for example, if there is low light, then it will decrease the rate of the photosynthesis. For example, if you have also a low carbon dioxide concentration, then the photosynthesis will decrease. So this is what's limiting factors. You have to know the list of limiting factors as well. This is only for the extended students. Now 6.2 is the leaf structure. There's no extra information for the extended students over here. It says, state that most leaves have a large surface area and are thin, and explain how these features are adaptations for photosynthesis. So over here, large surface area, this is memorization. There's adaptations for the plants and leaves. If there's large surface area, so that there is large amounts of carbon dioxide being diffused. As I said, there was the diffusion adaptations like in another chapter. I just explained it. If there's large surface area, then the diffusion will be like higher, and if it's thin, then there will be, of course, rapid diffusion because there's no like thick structures, so it will be much faster. Now you have to also explain the features like I said right now. So identify in diagrams and images the following structures in the leaf of a dichotomous plant, which means it has two seeds, that it has chloroplast, cuticle, guard cells, stomata, upper and lower epidermis, palisade mesophyll cell, and spongy mesophyll, air spaces, vascular bundles, which are made up of the xylem and phloem. So this is the structures that are in a plant. You have to know them, and this is very important that you have to know these stuff because they will ask you for their uses sometimes. And we also have to know that the palisade mesophyll layer, it has the largest amount of chlorophyll, and this is where the photosynthesis mainly takes place. And also we have to explain how the structures listed are adapted for photosynthesis, so you have to know this information as well. Chapter number 7 is about human nutrition. 7.1 is the diet, and both extended and core students, they have to know what is meant by a balanced diet. This is a definition, you can find it in the book. 2 says state the principal dietary sources and describe the importance of carbohydrates, fats and oils, proteins, vitamins limited to C and D, mineral ions limited to calcium and iron, fiber and water. You have to know where this, like for example, if you eat an orange, then your body will get vitamin C. This is the dietary source of vitamin C, for example. And you also know you also have to know what is the importance of proteins in your body, for example, and all of the things that are listed here. Now we also have to state the causes of scurvy and rickets. So scurvy is actually a deficiency of vitamin C, and rickets is caused by a deficiency in vitamin D. So you have to just know this like info. 7.2 is about the digestive system. Both core and extended students, they have to know and identify in diagrams and images the main organs of the digestive system, limited to alimentary canal, mouth, esophagus, stomach, small intestine, duodenum and ileum, large intestine, colon, rectum, and anus. So you have to, for example, if you get a picture in your exam in the paper 4, then you have to know, for example, if they ask you to label, you have to know where's the esophagus, where's the mouth, and you have to also know their uses. And you have to know about the associated organs like the salivary glands, pancreas, liver, and gallbladder. This is organs that are related to digesting and the digestive system. Number 2 says describe the functions of the organs that were listed in the 7.21, which is this part. And you have to know that ingestion is the taking in of substances, for example, food and drink into the body. So this is basically like definitions you have to know. Digestion is the breakdown of food. Absorption is the movement of nutrients from the intestine into the blood. Assimilation is uptake and use of nutrients by the cells. Digestion is the removal of undigested food from the body as feces. So all of these are definitions. You have to understand them as well, not just memorize them. Now for the 7.3 physical digestion part, the core students and extended students, they have to describe physical digestion as the breakdown of food into smaller pieces without chemical change into the food molecules. So this is a definition, you have to know it. Now the second part says state that physical digestion increases the surface area of food for the action of enzymes in chemical digestion. So this is basically why your body does physical digestion. It is to increase the surface area for food, and so the enzymes can actually do their job on the chemical digestion. So enzymes is actually chemical digestion, so you have to know this information, you have to memorize it. And 3 says identify in diagrams and images the types of human teeth: incisors, canines, premolars, and molars. So you have to look at an image and you have to identify where's the incisor, canine, premolar, and molars. You can find the diagrams in the book. The book is actually a really great resource to study from because you can look at images. If you don't have a book, then you can simply look at images online. So or you can ask your school teacher for help. Now, number 4 says describe the structure of human teeth limited to enamel, dentine, pulp, nerves, blood vessels, and cement, and understand that teeth are embedded in bone and gums. So you have to know that there's parts inside the teeth, like enamel. Enamel is the layer that is basically like the white part of your teeth. There's nerves and there's blood vessels. So, for example, if you eat something that is cold, then your teeth will like basically sense that it's cold, and it will send nerves to your brain. 5 says describe the functions of the types of human teeth in physical digestion of food. So, for example, incisors are for cutting food and molars are for grinding food. So, you have to know their uses. And number 6 says describe the function of the stomach in physical digestion. So, you have to just know what the stomach does in terms of digestion. Now, the extended students, they have an extra thing to know, which is number 7, outline the role of bile in emulsifying fats and oils to increase the surface area for chemical digestion. So there's something called bile. This bile is actually found in the stomach. It actually is used for emulsifying fats and oils, which means breaking down the oils and fats. The bile is actually just for the fats and oils. It exists so that it can increase surface area for chemical digestion. So this is like an enzyme. 7.4, which is about chemical digestion. All students, core or extended, they have to memorize that chemical digestion is the breakdown of large insoluble molecules into smaller soluble molecules. This is a definition you have to memorize. Number 2 says state the role of chemical digestion in producing small soluble molecules that can be absorbed. So the role or the use of chemical digestion, it exists so that that it can produce smaller molecules that your body can actually absorb into the blood, and that you can actually get. For example, if you eat something that is full of nutrients, your body can actually absorb the vitamins or the minerals that are in the food. This is done by chemical digestion. Number 3 says, describe the functions of enzymes as follows. Amylase breaks down starch to simple reducing sugars. So you have to know that this is the use of amylase, and amylase breaks down the starch. Proteases break down proteins to amino acids. Lipase breaks down fats and oils to fatty acids and glycerol. So this is basically like the uses and the names of enzymes that break down protein, starch, and fats and oils. Number 4 says state where in the digestive system amylase proteins and lipase are secreted and where they act. Number 4 says describe the functions of hydrochloric acid in gastric juice limited to killing harmful microorganisms in food, providing an acidic pH for optimum enzyme activity. So, as I said in chapter 5 of the enzymes, that there are some enzymes that actually work in like specific temperatures. So, and pHs. So, for example, there's an enzyme that may only work at pH 7 and then it gets denatured, and there's some enzymes that work at pH 2. These are actually found in the stomach, such as pepsin. They actually help the stomach in digesting and killing harmful microorganisms. So this is something that you need to know. Now, for extended students, they only this is only for the extended students, as I said. Describe the digestion of starch in the digestive system. Amylase breaks down starch into maltose. So actually, you have to know like specific information. There is more specific information for the extended students over here, that amylase doesn't break down starch into simple reducing sugars. It's actually called maltose for extended students. Maltase breaks down maltose to glucose on the membranes of the epithelium lining in the small intestine. So this is where the maltase actually works for extended students. You have to know this information. Now, number 7 says describe the digestion of protein by proteases in the digestive system. Pepsin breaks down protein in the acidic conditions of the stomach. So you have to know about pepsin. You have to know their names. These enzymes, they are present in the stomach, and pepsin is for breaking down protein, and trypsin is also in alkaline conditions on the small intestine. It breaks down protein as well. And so the difference between them is that pepsin works in acidic conditions and trypsin works in alkaline conditions. Number 8 says, explain that bile is an alkaline mixture that neutralizes the acidic mixture of food and gastric juices entering the duodenum from the stomach to provide a suitable pH for enzyme action. So this is basically a memorization sentence. You have to know it, and you also have to understand what it means. Core students and extended students have to know that the small intestine is where the nutrients are absorbed, and that the small intestine actually absorbs the most water from our body, but some of them is actually absorbed from the colon, so this is memorization. Extended students have to know about the villi and microvilli. It actually increases the surface area of the small intestine to help with the digestion process. And you have to also describe the structure of a villus. So if you have a figure of a villus, then you have to describe the different parts of it, and you also have to describe the roles of capillaries and lacteals in the villi. Number 8, which is chapter number 8, transport in plants. 8.1 is talking about the xylem and phloem. Core students have to state the functions of xylem and phloem. Xylem is actually for the transportation of water and mineral ions, and it also helps for support of the plant. Phloem is transportation of sucrose and amino acids. You have to also identify in diagrams and images the position of the xylem and phloem as seen in sections of roots, stems, and leaves of non-woody dicotyledonous plants, which means plants that have two seeds, as I explained before. So you have to be able to identify the positions of the xylem and phloem in different like areas or positions of an image. So for example, you have to know, if they get you a figure of the root, you have to know where the phloem and the xylem are, and if they get you a picture of the stem, then you also have to know where they are, and in the leaves you have to know. So you have to, of course, look at images and diagrams for this one. Now, for extended students, they have to relate the structure of the xylem vessels to their function, limited to thick walls with lignin. And basically, you have to know the different parts of the xylem vessel, and you also have to know their functions. Thick walls with lignin, they are basically for preventing the xylem from like bending over. So, for example, if you have a straw and you keep on like drinking from upwards over here, then the straw may actually bend. It may make something like this. So the lignin are actually for protecting the xylem from like bending over, and it may collapse. No cell contents, and they also have cells joined end to end with no cross walls to form a long continuous tube. Now for 8.2 is about water uptake. Core and extended students, they have to identify in diagrams and images root hair cells and state their functions. You have to look at root hair cells, and you have to see the different parts in there, and you have to also know their functions. State that the large surface area of root hairs increases the water uptake and mineral ions. So you have to like memorize this information, that large surface area helps the root hair cells, that they take more water and mineral ions. 3 says outline the pathway taken by water through the root, stem, and leaf as root hair cells, root cortex cells, xylem, and mesophyll cells. You have to know the order which the water comes out from the roots into the mesophyll cells. First of all, they go through the root hair cells, and then through the root cortex, and then through the xylem, and then it reaches the mesophyll cells where they are used for photosynthesis. Number 4 says investigate using a suitable stain the pathway of water through the above ground parts of a plant. Now for this part, there's no extra information for the extended students. 8.3, which is about transpiration. Core students have to describe transpiration as the loss of water vapor from the leaves, and actually for this part also extended students, they have to know about this. So this is basically a definition you have to memorize. This is very important. Now number 2 says state that the water evaporates from the surfaces of the mesophyll cells into the air spaces and then diffuses out of the leaves through the stomata as water vapor. So you have to know that this is how water evaporates from the mesophyll cells. Number 3 says investigate and describe the effects of variation of temperature and wind speed on transpiration rate. So if you have high temperatures and different wind speeds, then it may decrease or increase the transpiration rate according to the optimum for the transpiration. So if you have higher wind speeds, then the transpiration may increase. So now for the extended students, number 4 says explain how water vapor loss is related to the large internal surface area provided by the interconnecting air spaces between the mucosal cells and the size of and number of the stomata. So the large internal surface area actually it helps the air spaces so that the air can actually flow between the spongy mucosal cells to the palisade. So this is why they have large internal surface area, and explain the mechanism by which water moves upwards in the xylem in terms of a transpiration pull that draws up a column of water molecules held together by forces of attraction between water molecules. So water moves in the plant upwards, so this is actually against the gravity. It should actually move downwards, but there's a pull, that a force that helps the water to move upwards. This is by transpiration. So the water moves upwards against the gravity. By the help of transpiration, the water will evaporate and it will pull more water upwards. So this is how water moves in the xylem. Explain the effects on the rate of transpiration of varying the following factors: temperature, wind speed, and humidity. So there's different factors that may affect the rate of transpiration. They are stated over here, and you also have to know what happens if you increase the temperature. For example, what happens if you increase the wind speed, and if there's higher humidity, what will happen. Number 7 says explain how and why wilting occurs. 8.4 says translocation. There's no information for core students about this. This is only for extended students. Number 1 says describe translocation as the movement of sucrose and amino acids in the phloem from sources to sinks. So you have to describe, define this. In the exam, sometimes they will ask you to define translocation. Number 2 says describe sources as the parts of plants that release sucrose or amino acids. So this is basically definitions again. You have to describe, which means define, and sinks as the parts of plants that use or store sucrose or amino acids. So again, you have to be able to define this. This is basically memorization. Number 3 says explain why some parts of a plant may act as a source and a sink at different times. So sometimes in the plant, the root, in the winter, for example, when there is no sun, the root may actually break down some of the starch stores and provide energy for the rest of the plant. But sometimes the root in the summer will be actually a sink, which means it uses up the energy. So it will be a sink in the summer because it will be requiring energy from the photosynthesis that occurs in the summer. So different times may actually differ from being a source and a sink. So now chapter number 9, which is about transport in animals. 9.1, for core students and extended, they have to know describe the circulatory system as a system of blood vessels with a pump and valves to ensure one-way flow of blood. So this is again a definition, and you have to be able to memorize that. For extended students, you have to describe the single circulation of a fish. You have to know about it. Describe this double circulation of a mammal and explain the advantages of double circulation. Now for section 9.2, it is talking about the heart. In this part, the core and extended students, they have to know, identify in diagrams and images the structures of a mammalian heart limited to muscular wall, septum, left and right ventricles, left and right atria, one-way valves, and coronary arteries. So you have to be, if you're given a figure or if you're given a picture in the exam, you have to know where is the muscular wall, where is the septum, where is the ventricles located in the heart, and you have to be able to label the images. Now number 2 says state that the blood is pumped away from the heart in arteries and returns to the hearts in veins. So you have to know that the blood is pumped away from the heart in arteries, and then the veins bring back the blood from the rest of the body to the heart. And you have to know that the ECG is a way to monitor the activity of the heart, and that the pulse rate and listening to the pulse rate and listening to the sounds of the valves closing. This is actually when you have a doctor, he has this doctor, he or she, they have this little stethoscope. This thing actually, you when you put it on your chest, you listen to your heart's valves closing and opening. So this is basically the use of that stethoscope. Now 4 says investigate and describe the effect of physical activity on the heart rate. If you increase your physical activity, if you run for a long period of time, of course you will be breathless, you will be tired, and also your heart rate will increase so much because you are delivering blood more rapidly. Now 5 says describe coronary heart disease in terms of the blockage of coronary arteries, and state the possible risk factors including diet, lack of exercise, stress, smoking, genetic predisposition, age, and sex. So for example, sometimes the blockage of coronary arteries can occur if you have a high cholesterol diet, which means you're eating a lot of fried foods, for example, and lack of exercise. If you don't exercise a lot, of course, your coronary arteries may be blocked easily. Stress and smoking may cause also the coronaries to be blocked, which means the blood won't be able to pass through and reach the heart. So this is a big problem. Discuss the roles of diet and exercise in reducing the risk of coronary heart disease. So diet and exercise, of course, will help. You have to explain how does that actually reduce the risk of coronary heart disease, which occurs when the arteries that supply the blood to the heart are blocked. So this is the coronary heart disease. Now for extended students, they have to be able to identify in diagrams and images the atrioventricular and semilunar valves in the mammalian heart. So you have to know where they are located, and you have to be able to label the images. Number 8 says explain the relative thickness of the muscle walls of the left and right ventricles. So of course we know that the left ventricle is much thicker. It has a thicker muscle wall than the right ventricle, because the left ventricle actually it spreads out, or like it pumps the blood towards the aorta, which spreads out the blood to the rest of the body. Of course it has to be in high pressure so that it reaches all of the body parts. Now the muscle walls of the atria compared to those of the ventricles, the atria muscle walls are very thin compared to the ventricles, because the ventricles pump the blood towards the aorta and the lungs, so they have to be in high pressure. Explain the importance of the septum in separating oxygenated and deoxygenated blood. So the septum actually separates like the two parts of the heart. We know the heart is made up of two parts, the left and right side. The left side is always oxygenated, the right side is deoxygenated, because it collects blood from the body that is deoxygenated, and then the right ventricle actually pumps the blood towards the lungs, and then it comes back to the left ventricle and the left atria, where it is pumped towards the aorta, which then it is spread to other body parts. So this is basically the heart cycle. Describe the functioning of the heart in terms of contraction of the muscles of the atria and ventricles and the action of the valves. This is basically what I just explained, but you have to also know that the valves open and close at specific times. You can read more information in the book or in any notes. 11 says explain the effect of physical activity on the heart rate. If you, of course, increase your physical activity, if you do a lot of sports, then your heart rate will increase, because you are doing higher activity, which will cause the heart to work faster, to provide the blood faster for you, because you're doing a lot of physical activity. Now 9.3 is about the blood vessels. Core and extended students have to describe the structure of arteries, veins, and capillaries limited to relative thickness of wall, diameter of lumen, and presence of valves in veins. So you have to know that all of the arteries, veins, and capillaries, they have the things that are listed above. State the function of capillaries. You have to memorize that. And 3 says identify in diagrams and images the main blood vessels to and from the heart, limited to vena cava, aorta, pulmonary artery, and pulmonary vein. So you have to know that there are the blood vessels, they are vena cava, aorta, pulmonary artery, and pulmonary vein. Lungs, limited to pulmonary artery and pulmonary vein. So basically, these are like blood vessels that will carry things from the lungs and to the lungs. So pulmonary artery actually carries the blood away from the heart towards the lungs, and the pulmonary vein carries the blood from the lungs to the heart. Because as I explained above, the artery carries the blood away from the heart. Now, kidney, the renal artery supplies the blood towards the kidney, and the renal vein brings it back filtered blood to the heart. Now, for the extended students, they have to explain how the structure of arteries and veins is related to the the pressure of the blood that they transport. So, arteries may have thicker walls than veins because they have to withstand higher pressure. So veins don't have blood pressure in them like the arteries. And you have to also explain the structure of capillaries is related to how the structure of capillaries is related to their functions. You have to be able to identify in diagrams and images the main blood vessels to and from the liver as hepatic artery, hepatic veins, and hepatic portal vein. So there are blood vessels that carry blood to the liver and away from the liver. Hepatic artery carries away the blood from the liver, and the hepatic veins brings it back to the heart. So 9.4 blood. Core and extended students, they have to be able to list the components of blood as red blood cells, white blood cells, platelets, and plasma. So we have the blood, it is not just red blood cells, we have white blood cells as well, and platelets and plasma. 2 says identify red and white blood cells in photomicrographs and diagrams. You have to be able to know which part is, you have to be able to also label the image. 3 says state the functions of the following components of the blood. So there's functions, you have to memorize those. These are very important. Red blood cells in transporting oxygen, including the role of hemoglobin. You have to know what is the role of hemoglobin as well. White blood cells in phagocytosis and antibody production. Platelets in clotting. Details are not required for this one. Plasma in the transport of blood cells, ions, nutrients, urea, hormones, and carbon dioxide. So for platelets, you only have to state that they are used in clotting. In the old syllabus, we used to say the platelets process in clotting, but it is not required anymore in this new syllabus. You can check out my new syllabus changes. I made a video about that if you're entering exams from 2024 and upwards. Now, number 4 says state the roles of blood clotting as preventing blood loss and entry of pathogens. So you have to just say that blood clotting is a way to prevent blood loss and entry of pathogens. Now, for extended students, you have to identify lymphocytes and phagocytes in photomicrographs and diagrams. So you have to be able to know which one is lymphocyte and phagocytes when you are given a picture. Now, you also have to state the functions of lymphocytes. They are antibody productions. They are for antibody production, phagocytes. They are for engulfing pathogens by phagocytosis. So you have to just memorize that, and you have to also understand that lymphocytes are, as I said, for antibody production, and phagocytes are for engulfing pathogens, which means like eating the pathogens, like trapping them by phagocytosis. Number 7 says describe the process of clotting as the conversion of fibrinogen to fibrin to form a mesh. So you have to describe the process of clotting, and you also have to say the keywords of fibrinogen to fibrin, and that it forms a mesh that prevents further blood from leaking or preventing further blood. Now, chapter number 10 is diseases and immunity. 10.1 is talking about diseases and immunity. Core and extended students, they have to define a pathogen as a disease causing organism. Number 2, you have to also define transmissible disease as a disease which the pathogen can be passed from one host to another. A host is a person who is he is infected with the disease and then he can spread it to other people, then he is the host of that disease. Now state that a pathogen is transmitted by direct blood, including through blood. Through blood, you can get it from maybe a blood transfer, you can get it from the hospital if you're having anything related to blood. So other body fluids, maybe sneezing, maybe anything. So indirectly, you can also, a pathogen can get transmitted indirectly from contaminated surfaces. So for example, if you're sitting in a place and that table or chair, there was a contaminated person who was sitting on it, or maybe he touched it, then the bacteria can spread from one person to another through the surfaces, and also from food. So if someone sick was preparing your food, then you will have that disease if the bacteria can spread from that person to you. And also from animals, for example, if you have a pet that is sick at home, then that pet can actually spread the disease to you. And the air, so if someone is sneezing a lot in the air, then the bacteria will spread from one person to another through the air. Now 4 says describe the body defenses limited to skin, hairs in the nose. So skin can actually protect you from diseases because it protects your blood, and it protects your your internal organs. Now hairs in the nose, they actually trap the bacteria in your nose. Like instead of going directly into your nasal cavity, it will actually get trapped in the hairs of the nose. And mucus as well. Mucus is the sticky substance that is found in the throat and sometimes in the nose. This substance, it will actually trap again the bacteria and pathogens. Stomach acid, it breaks down and kills the bacteria inside because it is in acidic nature, which means it has acidic enzymes and it has acidic hydrochloric acid inside. So this will actually kill any bacteria by denaturing their enzymes. Number 5 is explain the importance of the following in controlling the spread of disease. A says a clean water supply. So people will need clean water to control or prevent the spread of diseases, because people will cook with this water and they may shower in that water. So we do not want diseases on our bodies, or we do not want to be eating from this in contaminated water. Now, hygienic food preparation. People need to wear masks when they are cooking in restaurants. And if at home, you have to be not sick when you are doing your food. And also, people in restaurants, again, they have to wear gloves. They have to always measure their temperature. If they have a flu or if they have a cold, then they don't. Good personal hygiene. This is very important so that you don't spread around diseases. And also water waste disposal, which means constantly cleaning your house, constantly throwing out the trash so that you don't attract bacteria inside your house. Sewage treatment, details are not required. So sewage treatment is treating the water from the sewage, removing the bacteria, chlorination. This stuff was required last syllabus, but now it's not required. Extended students, number 6, describe active immunity as the defense against a pathogen by antibody production in the body. So, immunity, this is another definition, active immunity. You have to be able to define this in the exam. State that each pathogen has its own antigens, which have specific shapes. So this is similar to the enzymes, but it's not it's the same concept. They have their own shapes, different shapes that only bind with specific antibodies. Now, describe antibodies as proteins that bind to antigens, leading to direct destruction of pathogens or marking of pathogens for destruction by phagocytes. Number 9, state that specific antibodies have complementary shapes which fit specific antigens. Number 10 says, explain that active immunity is gained after an infection by a pathogen or vaccination. So, active immunity, you can either get it by actually getting infected by someone or from a pathogen, or you can take the vaccination, which will still allow the pathogen to enter into your body, but um, it will have the same effects as the pathogen, but it will be in less concentration. Now, number 11, outline the process of vaccination. Weakened pathogens or their antigens are put into the body. So, this is what happens when you take vaccination. Weakened pathogens or their antigens are put into the body. So, they are weakened, as I said. So, also, their antigens stimulate an immune response by lymphocytes, which produce antibodies. So, this is what will give you the immunity later on. The antibodies will stimulate, as I said. Now, C says memory cells are produced that give long-term memory, immunity, I mean. So memory cells basically will know if you get the disease once again, then your body will already have a reaction done to it, so you will have immunity. It will not have an effect on you if you actually get the disease. Number 12 says explain the role of vaccination in controlling the spread of diseases. 13, explain that passive immunity is a short-term defense against a pathogen by antibodies acquired from another individual, including across the placenta and in breast milk. So you can get passive immunity, which is short-term. It is not long-term like the vaccination or actually getting the pathogen. So passive immunity is the opposite of active immunity. It is given from another individual, like across the placenta or in breast milk from the mother. So babies can have this so that they do not get infected and get easily sick and threaten their lives. Now, 14 says, explain the importance of breastfeeding for the development of passive immunity in infants. So you have to know that breastfeeding has some importance because it provides passive immunity. Now, state that memory cells are not produced in the passive immunity because memory cells are only in active immunity. Now, 10.1 continued. This is the another part that didn't fit in the first page. Extended students have to also know describe cholera as a disease caused by a bacterium which is transmitted in contaminated water. This is cholera is something that is spread only in contaminated water. So this is why we have to control and provide clean water to houses so that there is no cholera outbreaks. Explain that the cholera bacterium produces a toxin that causes secretion of chloride ions into the small intestine, causing osmotic movement, which means movement by osmosis of the water into the gut, causing diarrhea, dehydration, and loss of ions from the blood. So this is basically the process and the dangers of cholera bacterium. Chapter 11, which is about gas exchange in humans. 11.1, gas exchange in humans. Core students and extended, they have to describe the features of gas exchange surfaces in humans limited to large surface area, thin surface, good blood supply, and good ventilation with air. So you have to, if you want to have gas exchange in your body, or the optimum conditions for gas exchange, it is having large surface area so that there is large amounts of gas being exchanged, thin surface so that you can have lower diffusion distance, good blood supply to provide oxygen, and good ventilation to actually let out the carbon dioxide. Now, number 2 says identify in diagrams and images the following parts of the breathing system: lungs, diaphragm, ribs, intercostal muscles, larynx, trachea, bronchi, bronchioles, alveoli, and associated capillaries. So this is the breathing system organs. So you have to memorize those and know and also be able to label them in the exam. Number 3 says investigate the differences in composition between inspired and expired air using lime water as a test for carbon dioxide. So inspired air, which means the air that you breathe in, it will have higher oxygen composition. It basically has, I think, 21% of oxygen. So when you inspire out, it will have higher amounts of carbon dioxide and less oxygen. So this is the inspired and expired air. And you can demonstrate that change by lime water. Lime water is a test for carbon dioxide, and you should be able to show that in expired air it will have carbon dioxide, higher carbon dioxide. The lime water would change much faster in expired air, and inspired air it will take longer to change because it has lower carbon dioxide percentages. Number 4 says describe the differences in composition between inspired and expired air limited to oxygen, carbon dioxide, and water vapor. So, as I said, there is higher oxygen in inspired air and less oxygen in expired air. There is higher carbon dioxide in expired air and less in inspired air. And there's higher water vapor in the expired air. Now, number 5 says investigate and describe the effects of physical activity on the rate and depth of breathing. So, as I said before in chapter 9, that the physical activity will actually increase the rate and depth of breathing, and so you will breathe faster and more frequently when you are doing higher physical activities. Now, extended students, they have to know that you have to identify in diagrams and images the internal and external intercostal muscles. These are very important in the breathing process. Number 7 says state the function of cartilage in the trachea. 8 says explain the role of ribs, the internal and external intercostal muscles, and the diaphragm in producing volume and pressure changes in the thorax, leading to ventilation of the lungs. So, the internal and external intercostal muscles, they are the opposite. So if the internal intercostal muscle is contracting, then the external has to actually be relaxed. So these are the opposites, and the diaphragm will change shape according to whether you are breathing. Number 9 says explain the differences in composition between inspired and expired air. As I said before in the previous points, that the inspired air has higher oxygen than the expired air. Now, number 10 says explain the link between physical activity and the rate and depth of breathing in terms of an increased carbon dioxide concentration in the blood. So, when you do any vigorous activity, then there will be increased carbon dioxide concentration in the blood. This is because you're going to do higher respiration, and as we all know, that respiration it uses up glucose and oxygen, and it will produce carbon dioxide and water. So, there will be increased carbon dioxide in your blood, which is detected by the brain, leading to an increased rate and greater depth of breathing, so that you can allow your body to inhale more oxygen, so that your blood can have more oxygen. 11 says explain the role of goblet cells, mucus, and the ciliated cells in protecting the breathing system from pathogens and particles. So you have to also memorize this, and you also have to explain that if they ask you in the exam what is the role of goblet cells or mucus, you can say that they protect the breathing system from pathogens and particles by trapping in the pathogens. Now for 12, which is about respiration, chapter number 12, 12.1 respiration, core and extended students, they have to state the uses of energy in living organisms, including muscle contraction, protein synthesis, which means making of protein. So this is the use of energy in humans basically, or any living organism. Cell division, active transport. As I said, active transport requires energy. It is unlike osmosis and diffusion. Osmosis and diffusion is a passive process, which means they do not use energy. Growth, the passage of nerve impulses, and the maintenance of a constant body temperature. Why is it important that we have a constant body temperature? This is so the enzymes in our body can have their optimum temperature and optimum conditions, so that our body can work properly. Number 2 says investigate and describe the effect of temperature on respiration in yeast. So, temperature, if it increases too much, then there will be lower respiration because the denaturation of enzymes will occur. Now, if you decrease the temperature, then there will be low rate of respiration in yeast because it is not their optimum temperature. 12.2, which is about aerobic respiration, describe aerobic respiration as the chemical reactions in cells that use oxygen to break down nutrient molecules to release energy. So, aerobic respiration and anaerobic respiration, what is the difference between them? Aerobic respiration uses up oxygen, and anaerobic uses up no oxygen. So, over here you have to also be able to define this. This is a definition, and you also have to understand it. Now, what is more important, that you have to know the word equation for aerobic respiration as glucose plus oxygen, which produces carbon dioxide and water. This is the main thing that happens in human body when you are doing exercise, as I just explained in the previous chapter. Then you will have carbon dioxide and water, which will cause your body to breathe faster because you have higher carbon dioxide in your body. Now, for extended students, the core students only have to know the word equation. Now, for extended students, you have to know the balanced chemical equation, which is this one, C6H12O6 plus 6 oxygens. It will produce 6 carbon dioxide and 6 water molecules. So this is basically the opposite of the photosynthesis equation. So you have to memorize that, and you also have to write it down as a balanced chemical equation. Anaerobic respiration, which is 12.3, core students have to describe anaerobic respiration as the chemical reactions in cells that break down molecules to release energy without using oxygen. So this is the same definition as the aerobic respiration, but you say without the using of oxygen. Now, number 2 says, state that the anaerobic respiration releases much less energy per glucose molecule than aerobic respiration. So, anaerobic respiration, it does not really release that much energy, like compared to aerobic respiration. So, we have to know that this piece of information. Now, number 3 says, state the word equation for anaerobic respiration in yeast. As glucose, it will produce alcohol and carbon dioxide. So anaerobic respiration, it occurs in yeast only, and some bacterias. It will take the glucose and it will produce alcohol and carbon dioxide. So this is the fermentation process. If you take chemistry, you already are familiar with this. Number 4 says, state the word equation for anaerobic respiration in muscles during vigorous exercise as glucose, and it will produce lactic acid. So actually in humans, they have anaerobic respiration as well, in muscles only, when you are doing vigorous exercise, which is like hard or high intensity exercises. It will convert glucose to lactic acid. This is when it will not find any oxygen in your blood, then it will refer, or it will go to make it will break down energy so that you can be able to still move, and it will produce lactic acid, which will actually hurt, and it will make you stop because you have to take breath, you have to take oxygen into your body so that you can continue to respire, because this is like the last option for your body to remain breaking down energy. Number 5, which is for extended students only, state the balanced chemical equation for anaerobic respiration in yeast as C6H12O6, which will produce 2C2H5O5, which is the chemical equation for alcohol, and this is carbon dioxide, 2CO2. So you have to memorize this as well. Number 6 says, state that the lactic acid builds up in muscles and blood during vigorous exercise, causing an oxygen debt. Now, oxygen debt is when your body needs oxygen, but you do not provide oxygen. So it will go and it will actually, as I said, it will break down glucose to lactic acid so that you can continue to move and your body will still provide energy. But then you have to sit down after your exercise and just breathe so that your body can provide oxygen again in normal supply to the cells of your body. Now, 12.3 continued. Supplement students, they have to outline how the oxygen debt is removed after exercise limited to continuation of fast heart rate to transport lactic acid in the blood from muscles to liver. So after you finish your exercise, this is how your oxygen debt is removed. First of all, you're going to have a fast heart rate so that your body can rapidly remove the lactic acid from the blood to the liver so that it can be broken down. You have to also, your body will continue to have deep and fast breathing rates, to supply oxygen for aerobic respiration of lactic acid. So after that, it will be aerobically respired in your liver. This is how the lactic acid will be broken down. And then the aerobic respiration of lactic acid in the liver, as I just said, So this is how oxygen debt is removed in 3 steps. You have to memorize this and understand it. Now, excretion. This is chapter 13 in humans. Core and extended students, they have to state that carbon dioxide is excreted through the lungs. You also have to state that the kidneys excrete urea and excess water and ions. So this is where excretion mainly occurs in humans. 3 says identify in diagrams and images the kidneys, ureters, bladder, and urethra. So you have to be able to label and be able to identify those structures in pictures. Now, for extended students, number 4, identify in diagrams and images the structure of the kidney limited to the cortex and medulla. I have just actually uploaded a video of May-June 2023 solving paper 2. I have actually solved a question that is related to this cortex and medulla. You can check it out over there. Number 5 says outline the structure and function of a nephron and its associated blood vessels limited to the role of glomerulus in the filtration from the blood of water, glucose, urea, and ions. So actually we have glomerulus in our kidneys, and these have the job or the role of filtering the blood from water, glucose, urea, and ions. We have also the nephron, which is it helps in the reabsorption of all the glucose and some of the ions and most water back into the blood. So this will help in the reabsorption again. Now, formation of urine containing urea, excess water, and excess ions. Details of these processes are not required. Last syllabus, we used to be able we used to have the details of these processes, but they are removed in the new syllabus. As I said in my new syllabus changes video, you can check it out for more details. Number 6 says describe the role of the liver in the assimilation of amino acids by converting them to protein. So the liver, if they ask you in the exam, you have to say that it has the role of assimilation of amino acids by converting them to proteins. Number 7 says state that the urea is formed in the liver from excess amino acids. You also have to describe deamination as the removal of nitrogen containing part of amino acids to form urea. So this is basically the deamination definition. You have to memorize this one. And explain the importance of excretion limited to the toxicity of urea. So excretion is important so that you can remove the toxic urea from your body, because if it's kept in your body, then it will be toxic for you and for your blood. Chapter 14, which is about coordination and response. 14.1, core students and extended students, they have to state that electrical impulses travel along neurons. Number 2 says describe the mammalian nervous system in terms of the central nervous system and the peripheral nervous system. So there's 2 parts, which is the CNS and PNS. And the CNS is made up of the brain and the spinal cord, and the PNS is made up of nerves outside of the brain and spinal cord. So now for the number 3 says describe the role of the nervous system as coordination and regulation of body functions. Number 4 says identify in diagrams and image sensory relay and motor neurons. So you have to be able to know and label the images if they ask you for the sensory relay and motor neurons. Now, number 5 says describe a simple reflex arc in terms of receptor, sensory neuron, relay neuron, motor neuron, and effector. So there's these structures inside of the reflex arc, simple reflex arc. Describe a reflex action, which this is a definition, by the way, as means of automatically and rapidly integrating and coordinating stimuli with the responses of effectors, muscles, and glands. So reflex action actually happens automatically, and this is by the help of stimuli, coordination of stimuli, and the response of effectors, which is the muscles and glands. Effectors are like the structures that will help like do the action. So for example, if you are required to move, then the effectors are muscles. It will help you to move. If you want to, for example, if your body needs to sweat, then it will actually like send stimuli to the sweat glands so that your body can sweat. So these are effectors. Number 7 says describe a synapse as a junction between 2 neurons. This is another definition you have to memorize. Now for extended students, They have to know, describe the structure of a synapse, including the presence of vesicles containing neurotransmittal molecules, the synaptic gap, and receptor proteins. Number 9, describe the events at a synapse as an impulse stimulates the release of neurotransmittal molecules from vesicles into synaptic gap. The neurotransmitter molecules diffuse across the gap. Neurotransmitter molecules bind with the receptor proteins of the next neuron. An impulse is then stimulated in the next neuron. So this is the events that happen at a synapse, and they're very important. Number 10 says state that synapses ensure impulses travel in one direction only. So this is the use of synapses in our nervous system. Now for sense organs, 14.2, core students and extended students, you have to describe sense organs as a group of receptor cells responding to specific stimuli, light, sound, touch, temperature, and chemicals. So this is a definition. Number 2 says identify in diagrams and images the structures of the eye limited to cornea, iris, pupil, lens, retina, optic nerve, and the blind spot. So these are structures and like parts of the eye that you have to know, and you also have to be able to label those when you are needed to. Describe the function of each part of the eye limited to cornea, refracts light, iris controls how much light enters the pupil, lens focuses light on the retina, retina contains light receptors, some are sensitive to light of different colors, and optic nerves carries impulses to the brain, and so this is the uses of each part of the eye as I just explained. 4 says explain the pupil reflex limited to changes in the light intensity and pupil diameter. So for example, the pupil diameter may increase depending on the light intensity that you're in, so the pupil may become bigger, the black part of your eye. Now, number 5, explain the pupil reflex in terms of antagonistic action of circular and radial muscles in the iris. Explain recommendation to view near and distant objects in terms of the contraction and relaxation of the ciliary muscles, tension in the suspensory ligaments, and shape of the lens and refraction of the light. You also have to know the distribution of rods and cones in the retina of a human. So you have to know where are the rods and cones in our eye. The outline that the function of rods and cones limited to greater sensitivity of rods for night vision. So rods are basically responsible for your night vision, which is like black and white, and the cones are responsible for absorbing light of different colors for the color vision. So cones are for color vision, and they provide red, green, and blue colors so that you can see those colors. Identify in diagrams and images the position of the fovea and state its function. So you have to know where the fovea is, and you also know the function. You have to know the function of the fovea. Moving on to 14.3, which is about hormones, you have to define hormone as a chemical substance produced by a gland and carried by the blood, which alters the activity of one or more specific target organ, and identify in diagrams and images specific endocrine glands and state the hormones they secrete limited to adrenal glands, which produce adrenaline. Pancreas, they produce insulin. Testes, they produce testosterone. Ovaries and estrogen, so that means that ovaries, they will produce the estrogen. Describe adrenaline as the hormone secreted in the fight or flight situations, and its effect limited to increased breathing rate. So when there's this hormone in your body, then it will cause your body to actually increase the breathing rate, and it will also increase the heart rate, and it will increase the pupil diameter. So that means your eye, the black part of your eye, which is called the pupil, it will increase in size, it will be wider and bigger. Compare nervous and hormonal control limited to the speed of action and duration of effect. So nervous system is much faster than the hormonal system, because nervous system, because it has the impulses and the things, but hormonal control is much slower than the nervous system, because there is glands and there is hormones to be secreted, and these take time to actually like work and have their effects. State that glucagon is secreted by the pancreas, so you have to know that the pancreas actually produces the glucagon. Number 6, describe the role of adrenaline in the control of metabolic activity, limited to increasing the blood glucose concentration and increasing the heart rate. So extended students have to know these extra points other than the ones that the core students know. Moving on to 14.4, which is about homeostasis. Core and extended students have to describe homeostasis as the maintenance of constant internal environment. And you have to also state that insulin decreases blood glucose concentration. Moving on to the extended part, explain the concept of homeostatic control by negative feedback with reference to a set point. So, for example, the body will keep itself warm, so it has to be at a specific temperature, or else there will be consequences. Describe the control of blood glucose concentration by the liver and the roles of insulin and glucagon. So, you have to know that insulin decreases the blood glucose concentration, and glucagon actually increases the blood glucose concentration. Outline the treatment of type 1 diabetes, identifying diagrams and images of the skin, hair erector muscles, sweat glands, receptors, sensory neurons, blood vessels, and fatty tissue. So when you're given a diagram, you have to be able to label and know. Describe the maintenance of constant internal body temperature in mammals in terms of insulation, sweating, shivering, and the role of the brain. So, maintenance of a constant internal temperature may be done by the several ways by insulation, so that, for example, if an animal is in a very cold environment, then it will have thick layer of fat tissues around their internal organs so that they can protect themselves. Sweating, maybe sweating if you are in a hot environment, then if sweating will help your body cool down. Shivering, so that it will release heat energy and that your body will keep cool, keep warm. Role of the brain, of course, to transmit impulses, as I just explained in chapter 14. Number 8, describe the maintenance of a constant internal body temperature in mammals in terms of vasodilation, which is making the arterioles become wider and supply more blood to help you keep warm. Vasoconstriction is when the body will actually may narrow down the arterioles so that there is less blood supply. So these actually supply the skin surface capillaries. 14.5 Tropic responses. Core and extended students have to define gravitropism as a response in which parts of the plant grow towards or away from gravity, and describe or define phototropism as a response in which parts of the plant grow towards or away from the direction of the light source. Number 3 says to investigate and describe gravitropism and phototropism in shoots and roots. So you have to be able to describe gravitropism. As I just said over here, you can say this statement. And also the same applies to phototropism. You can write down this sentence to describe what is really phototropism. Now for extended students, they have to explain phototropism and gravitropism of a shoot as examples of chemical control of a plant growth. So these are examples of chemical control of a plant growth. Sometimes the plant will have chemicals within itself to help control its plant growth. Sometimes it is external factors that will help the plant grow or not. Number 5, explain the role of auxin in controlling shoot growth. Limited to auxin is made in the shoot tip. Auxin diffuses through the plant from the shoot tip. So auxin, auxin, as it's written down, it is actually helping the plant control its growth, and it's also auxin is unequally distributed in response to light and gravity, so it is actually produced according to the light and gravity that is around the plant. Auxin stimulates cell elongation, which means it will help the cells live longer. Now, chapter 15 is about drugs. So this is a quite short chapter compared to last syllabus. It was quite long, the last syllabus. It was longer than this, as I remember. Describe a drug as any substance taken into the body that modifies or affects chemical reactions in the body. So this is the definition of a drug. It is anything that is not made by your body. It is actually man-made, and you take it in to affect the chemical reactions that naturally occur in your body. Describe the use of antibiotics for the treatment of bacterial infections. So you have to know that we can use antibiotics for the treatment of bacterial infections only. You don't use it for viral infections, which are caused by viruses. Now, number 3 says state that some bacteria are resistant to antibiotics, which reduces the effectiveness of antibiotics. So sometimes the antibiotics will not work because your body already has like kind of an immune system, or it's immune to it because the bacteria are resistant to the antibiotics. So you have to look for other alternatives. State that antibiotics kill bacteria but do not affect viruses, as I just explained in the other point. Number 5, which is only for extended students, explain how how using antibiotics only when essential can limit the development of resistant bacteria such as MRCA. So basically, you only have to use antibiotics when you only need it, because sometimes you will have resistant bacteria developing inside of your body. This will make it hard to treat any kind of bacterial infection in the future. Now 16, which is reproduction. 16.1 is about asexual reproduction. Describe asexual reproduction as the process resulting in the production of genetically identical offspring from one parent. So asexual reproduction is from one parent only, and this is the opposite of sexual reproduction, which I'm going to be talking about later on. So this is the definition. You have to be able to define this in the exam and memorize it. Identify examples of asexual reproduction in diagrams, images, and information provided. Now, for extended students, they have to discuss the advantages and disadvantages of asexual reproduction. Now, there's an advantage of asexual reproduction, which is to increase the population. It can actually increase the population of a species, and it can increase the crop production. But there's disadvantages actually, which is maybe getting a disease. Asexual reproduction does not have like genetic difference. All of the species are having the same genes. So if there's one disease that can wipe out one of them, then all of the crops will be destroyed because they all are the same. So this is a problem. 16.2 is talking about sexual reproduction. Core and extended students have to define sexual reproduction as a process involving the fusion of the nuclei of two gametes to form a zygote, and the production of offspring that are genetically different from each other. So this is, as I said, the opposite of asexual reproduction. It involves two gametes, it involves two parents. So the gametes are actually from either the testes or from the ovaries. They will form a zygote by the fusion of both of their nuclei, and they will produce offspring that are genetically different, not genetically the same. Describe fertilization as the fusion of the nuclei of gametes. So this is another definition. Now, for extended students, you have to state that the nuclei of gametes are haploid, which means they only have one gene inside, and the zygote is diploid, which means it has two genes inside, two pairs of chromosomes, and the haploid has only one pair. So for example, if you're going to mate, then there is one haploid and one haploid. This will produce two pairs. So this is the normal human chromosome, like diploid. So it can produce diploid zygote, not four. If there's two and two that will actually mate, then it will produce four, which is not normal for the human chromosomes. So now discuss the advantages and disadvantages of sexual reproduction to a population of the species in the wild. So advantages is that there will be genetic differences which will cause the population to be more resistant to a disease. Crop production, the same thing applies to the crops. Now, sexual reproduction in plants, 16.3, core and extended students, they have to identify in diagrams and images and draw the following parts of an insect pollinated flower: sepals, petals, stamens, filaments, anthers, carpels, style, stigma, ovary, and ovules. So these are the parts of the plant in sexual reproduction. Now state the functions of the structures listed in 16.3.1. So you have to state their functions as well. So 3 says identify in diagrams and images and describe the anthers and stigmas of a wind-pollinated flower. So you have to be able to label the images of a wind-pollinated flower, and you have to know where each of these structures are located. 4 says distinguish between the pollen grains of insect-pollinated and wind-pollinated flowers. So you have to be able to know the difference between pollen grains from the insect-pollinated and the wind-pollinated flowers. Describe pollination as the transfer of pollen grains from anther to stigma. So this is a definition. You have to be able to memorize that. For extended students, you have to describe self-pollination as the transfer of pollen grains from an anther of a flower to the stigma of the same flower or a different flower on the same plant. So self-pollination occurs in one flower or inside one plant. So a plant can have several flowers on it. So it may transfer stigma. It may transfer pollen grains from the anther to the stigma on the same flower. So this means that the flower has a male flower and a female flower, and they can both pollinate each other. So this is self-pollination, all within the same plant. Now for cross-pollination, which is the transfer of pollen grains from the anther of a flower to the stigma of a flower on a different plant of the same species. So cross-pollination is the transfer of pollen grains from different plants, from different flowers from different areas, not on the same plant. So this is the difference. Discuss the potential effects of self-pollination and cross-pollination on a population in terms of variation, capacity to respond to changes in the environment, and reliance on pollinators. So self-pollination actually can increase the population of a plant very rapidly if it's in an isolated area where there is no more plants. So this is an advantage of self-pollination, and cross-pollination may actually deplete their population if there is no flowers around. So if that plant dies, then that species can actually become extinct. Now also they are reliant on pollinators so that they can survive. 16.3 Sexual reproduction in plants continued. Core students have to state that fertilization occurs when a pollen nucleus fuses with a nucleus in an ovule. Describe the structural adaptations of insect-pollinated and wind-pollinated flowers. So you have to be able to describe the adaptations of both insect-pollinated and wind-pollinated. Investigate and describe the environmental conditions that affect germination of seeds, limited to the requirement for water, oxygen, and a suitable temperature. So germination is basically the growth of seeds inside of the roots, inside of the soil. So when the seed is ready, then it will grow out a plant, it will grow out the fruit, it will grow out the flower in the future. So germination is very important so that the plant can have roots down. Germination only needs water, oxygen, and a suitable temperature so that it can occur. Now, supplement students, extended students, they have to describe the growth of pollen tube and its entry into the ovule, followed by fertilization. Details of the production of endosperm and development are not required. So you only have to describe the growth of pollen tube. So the pollen tube is done in the stigma so that it can allow the gametes to enter into the ovule inside of the plant so that the fertilization can occur. 16.4 Sexual reproduction in humans. Now this is a quite long part, so you have to focus right here because this is a very important chapter. It comes very frequently in all exams. Core students and extended students, they have to identify on diagrams and state the functions of the following parts of the male reproduction system: testes, scrotum, sperm ducts, prostate, gland, urethra, and the penis. So these are the parts of the male reproductive system in the humans. Now, number 2 says identify on diagrams and state the functions of the following parts of the female reproduction system, which is ovaries, oviducts, uterus, cervix, and the vagina. Number 3 says describe fertilization as the fusion of nuclei from a male gamete sperm and a female gamete egg cell. So this is the definition of fertilization for humans. Number 4 says explain the adaptive features of sperm limited to flagellum, mitochondria, and enzymes in the acrosome. So the sperm cell actually has structures within it. It has flagellum, just like the bacteria, so that it can rapidly move around inside the ovary, and also the mitochondria, so that it can release energy for its movement, and enzymes in the acrosome to break down the jelly coating around the egg cell so that they can fertilize. Explain the adaptive features of egg cells limited to energy stores and the jelly coat that changes to at fertilization. So energy stores, so that the egg cell can continue to live, and also the jelly coat that I just explained, it is protecting the egg cell, so it will actually break down according to the sperm cell that has just entered. It will only allow one sperm cell at a time, so there is no way there will be two, or it can occur in which the twins will be born. So this is how twins are born, and it's very rare, it's not something that happens frequently. Number 6 says compare female and male gametes in terms of size, structure, mortality, and numbers. So male gametes, they are actually much higher in quantity than the female gametes. They are also having a very bacterial kind of structure. It is something like that, and it also have the acrosome at the front. This is to again break down to the jelly coat in the egg cell. And also they have higher numbers and they are very fast, unlike the female gametes that do not move in its position. Number 7 says state that in early development the zygote forms an embryo, which is a ball of cells that implants into the lining of the uterus. So in early development, the zygote, which is the baby, it will form an embryo, which is a ball of cells that implants into the lining of the uterus, as I just said. So you have to understand this piece of information. Number 8, identify on diagrams and state the functions of the following in the development of the fetus: umbilical cord, placenta, amniotic sac, and amniotic fluid. So these are the structures that are found in the developing fetus. Number 9, which is only for extended students, describe the function of the placenta and umbilical cord in relation to the exchange of dissolved nutrients, gases, and excretory products between the blood of the mother and the blood of the fetus. So the placenta is actually very important, and the umbilical cord, so that the mother can provide energy, provide nutrients and gases, oxygen for the growing fetus so that it cannot die. And the mother will actually take the excretory products from the fetus and it will excrete it. And also the blood of the fetus is also exchanged between the mother and the fetus. State that some pathogens and toxins can pass across the placenta and affect the fetus. So also the placenta can let in toxins and pathogens, and this is very dangerous. So this is why the mother has to take good care of herself during pregnancy. 16.5 Sexual hormones in humans. Core and extended students have to know and describe the roles of testosterone and estrogen in the development and regulation of secondary sexual characteristics during puberty. So you have to know that testosterone and estrogen, they are basically very important of the secondary sexual characteristics. So this helps the children actually go through puberty, so you have to know about this. Describe the menstrual cycle in terms of changes in the ovary and in the lining of the uterus. Now for extended students, number 3, describe the sites of production of estrogen and progesterone in the menstrual cycle and in pregnancy. So you have to know where the estrogen and progesterone are made, and you also you have to know that the estrogen and progesterone are not just used in the menstrual cycle, but they are also used in pregnancy when women are pregnant. Now number 4 says explain the role of hormones in controlling the menstrual cycle and pregnancy limited to FSH, LH, progesterone, and estrogen. So you have to know the uses of FSH, LH, and how they control progesterone and estrogen, and how they control the menstrual cycle and pregnancy. And 16.6 is sexually transmitted infections. Core and extended students, they have to know and describe a sexually transmitted infection, STI, as an infection that is transmitted through sexual contact. So this is a definition, you have to know it. Number 2 says state that human immunodeficiency virus HIV is a pathogen that causes an STI. You have to know this. And number 3 says state that HIV infection may lead to AIDS. AIDS is very serious and it can take years to know that you actually have AIDS, and it's really life-threatening disease. Number 4 says describe the methods of transmission of HIV. Number 5 says explain how the spread of STIs is controlled. So you have to know these methods. All of this is both for extended and core students. 17, chapter 17, inheritance. 17.1 is chromosomes, genes, and proteins. Core and extended students, they have to state that chromosomes are made of DNA, which contains genetic information in the form of genes. You have to memorize this information because sometimes you're asked about this in the exam. Number 2 says define a gene as a length of DNA that codes for a protein. This is a definition, of course. Number 3 is also a definition. Define an allele as an alternative form of a gene. 4 says describing the inheritance of sex in humans with the reference of X and Y chromosomes. So a male will actually have XY chromosome. A female, on the other hand, will have only XX chromosomes. So you have to know this. This is very important. Extended students, they have to know about number 5. State that the sequence of bases in a gene determines the sequence of amino acids used to make a specific protein. Knowledge of the details of nucleotide structure is not required. Number 6 says explain that different sequences of amino acid give different shapes to protein molecules. Here's the continuation. Supplement students, extended students, they have to explain that DNA controls cell function by controlling the production of proteins, including enzymes, membrane carriers, and receptors for neurotransmitters. So all of these are mainly memorization information, but these are very important, and this is one of the most important chapters along with chapter 16, because these are the most chapters that I've seen in the exams. So I'm not saying all of the syllabus is not important, but these are very important and you have to focus on these a lot. Explain how a protein is made, limited to the gene coding for the protein remains in the nucleus. Messenger RNA, mRNA, is a copy of a gene. mRNA molecules are made in the nucleus and move to the cytoplasm. The mRNA passes through ribosomes. The ribosome assembles amino acids into protein molecules. The specific sequence of amino acids is determined by the sequence of bases in the mRNA. You are not required to know about transcription or translation. So this is the very, very important. You have to memorize all of this. You have to also understand it. This is the process of how a protein is made. As I said in the previous chapters, that energy is used to make protein, and also the protein is made in the nucleus. So these are important things you have to know about this process. Number 9, explain that most body cells in an organism contain the same genes, but many genes in a particular cell are not expressed because the cell only makes the specific proteins it needs. Number 10 says, describe a haploid nucleus as a nucleus containing a single set of chromosomes. So this is a definition you have to know, and you also have to understand that because this is important when you're solving genetics questions. 11 says, describe a diploid nucleus as a nucleus containing 2 sets of chromosomes. So during gametes, when they actually bind, there is only, the gametes are haploid. These are the only cells that are haploid. They are called sex chromosomes. When they bind, they only have a single set of chromosomes. So when they bind together, they will form double, like I explained before in another chapter. So this is why they are haploid, so that you don't get 4 sets in the end. You have to get 2 sets because this is the normal human genes. Describe a diploid nucleus as a nucleus containing 2 sets of chromosomes. 12 says, state that in a diploid cell there is a pair of each type of chromosome, and in a human diploid cell there are 24 pairs. So now for 17.2, which is about mitosis. This part is only for extended students, so if you're a core student, you don't need to know about mitosis. Extended students have to describe mitosis as a nuclear division giving rise to genetically identical cells. The stages are not required. Now, this is another definition. State the role of mitosis in growth, repair of damaged tissues, replacement of cells, and asexual reproduction. You have to also know about the exact replication of chromosomes occurs before mitosis. State that during mitosis, the copies of chromosomes separate, maintaining the chromosome number in each daughter cell. Describe stem cells. So this is another definition. Stem cells are unspecialized cells that divide by mitosis to produce daughter cells that can become specialized for specific functions. So this is the rest of it. State that meiosis is involved in the production of gametes. So mitosis is only for diploid cells, and meiosis is different. It is for actually the production of gametes. It is for the division of gametes. Meiosis as a reduction division in which the chromosome number is halved from diploid to haploid, resulting in genetically different cells. So these are also, this is a definition you have to know about. 17.4 monohybrid inheritance. Both extended and core students have to know about this. Describe inheritance as the transmission of genetic information from generation to generation. This is another definition. Describe genotype as the genetic makeup of an organism and in terms of alleles present. Another definition. 3 and 4 are also definitions. Describe phenotype as the observable features of an organism. So phenotype is basically how the organism looks like, and genotype is the genetics of the organism from inside. You don't see it, of course. Describe homozygous as having 2 identical alleles of a particular gene, and state that 2 identical homozygous individuals that breed together will be pure breeding. And describe heterozygous as having 2 different alleles of a particular gene. So all of these are actually keywords. You have to know them very well and understand what they mean, because these words will give you marks in the exam just by mentioning the word genotype or phenotype or homozygous. You may get marks in the exam, so please be careful with these keywords. Another definition. Describe heterozygous as having 2 different alleles of a particular gene. State that a heterozygous individual will not be pure breeding. Pure breeding is only for homozygous individuals, which means they have that same, for example, H and H capital. This is heterozygous, which means they have different alleles. One is the dominant and one is the recessive. Dominant is an allele that is expressed if it is present in the genotype. So it is basically, dominant allele is like the capital letter and the recessive is like the small letter. So this is the difference. This is heterozygous because it has both dominant and recessive. Homozygous can be having 2 dominant or having 2 recessive. So you can also say that, or heterozygous is having different types of allele. Recessive allele as an allele that is only expressed when there is no dominant allele of the gene present in the genotype, as I just said. So again, definitions. Interrupt p-degree diagrams for the inheritance of a given characteristic. These are only found in paper 4. So you have to solve a lot of paper 4 questions to really get to know about this diagrams. 11 says use genetic diagrams to predict the results of monohybrid crosses. Calculate phenotypic ratios limited to 1:1 and 3:1 ratios. So 1:1 is basically like half-half, 50% and a 50%. And 3:1 means that there's 3 and 1, for example, recessive. So you have to know about the monohybrid crosses. I have been doing paper 2 videos. I have been also paper 4 videos in some of them. I've solved some questions with the monohybrid crosses and different ways to actually like do these questions. So if you're in doubt, you can also watch my solving videos. I've done this monohybrid crosses a lot. 12 use a Punnett square in crosses which result in more than one genotype to work out and show the possible different genotypes. Again, I also work with Punnett squares, which are like this. You basically put, like, for example, A and a recessive, and then you put like this. So this is how I basically solve. You can, as I said, check out my videos if you have any questions about that. You can see how I solve that. 13 is only for extended students. Explain how to use a test cross to identify an unknown genotype. And the test cross is like, for example, like this, and you have this one. You can always cross them like this. So this is crossing, and this is the Punnett square. Sometimes they don't really ask you to use a specific way. You can use whatever way you find easier. Monohybrid continued. Describe codominance as the situation in which both alleles in a heterozygous organisms contribute to a phenotype. Another definition. Explain the inheritance of A, B, O blood groups. Phenotypes are A, B, AB, and O blood groups, and the alleles are I, A, I, B, and I, O. Describe a sex-linked characteristic as a feature in which the gene responsible is located on a sex chromosome, and this makes the characteristic more common in one sex than in the other. Again, you have to understand this. 17. Describe red-green color blindness as an example of sex linkage. 18 says use genetic diagrams to predict the results of monohybrid crosses involving codominance or sex linkage. Chapter 18, which is about variation and selection. For core and extended students, you have to know about this definition. Variation as species between individuals of the same species. Differences, I mean, between individuals of the same species. State that continuous variation results in a range of phenotypes between two extremes. Example include body length and body mass. So there is variation, of course, between each and every human. There is variation. There is extremes of body length and body mass. So for example, you can be too tall or you can be too short. This is extremes. And then in the middle, for example, this is like the average height. This is not an extreme. So there is, of course, variation between people in these types of stuff. So this is an example of continuous variation. Now, this continuous variation results in a limited number of phenotypes with no intermediates. Example include ABO blood groups, seed shape in peas, and seed color in peas. So there is always stuff there that there is no discontinued variation is when there is no there's no variation in this stuff, such as the blood groups. You can be either blood group from A, or you can be B, or you can be like mixed blood group. So there is no variation. You cannot get like a new blood group. And also the seed shape, like you can either, like for example, the popcorn seeds, they are always in the same shape. There is no any variation ever. So state that discontinuous variation is usually caused by genes only, and continuous variation is caused by both genes and the environment. So what does this mean? Discontinuous variation is usually caused by genes only. So for example, the blood group, you can only inherit this feature, but continuous variation, for example, your height, your body height as a human, you can always like environmental conditions, such as if you eat good nutrition, if you have a healthy diet, then you may grow taller than others. And also sometimes your skin color can be affected. For example, when you go to the beach, your skin color can get darker and it can get lighter sometimes according to the place you're in. So this is environmental conditions. Number 5 says investigate and describe examples of continuous and discontinuous variation, like I just said. Number 6 says mutation. Describe mutation as genetic change. Number 7 says state that mutation is the way in which new alleles are formed. So mutation is like something that is not normal that happens in the genes. So for example, Down syndrome. Down syndrome, as we all know, it is when there is an extra copy of a chromosome, and this will cause like not normal human like chromosomes. They will have an extra pair of chromosomes. So this is mutation. It is not normal. Not everyone is like that. So this is how new alleles are formed, and sometimes they can pass those mutated alleles to another individuals. State that ionizing radiation and some chemicals increase the rate of mutation. So radiation and some chemicals can cause a mutation. Now for extended students, they have to describe gene mutation as random change in the base sequence of DNA. state that mutation, meiosis, random mating, and random fertilization are sources of genetic variation in populations. Now, 18.2 is about adaptive features. Core and extended students have to describe adaptive feature. This is another definition as an inherited feature that helps an organism to survive and reproduce in its environment. Now, number 2 says interrupt images or other information about a species to describe its adaptive features. So, for example, polar bears that live in the very cold areas in the world, such as Antarctica, these have you always find the polar bears, they are not they have a lot of fur, and they're also having a lot of they're sometimes really chubby. So this is not because they eat a lot. This is because their genes, they always turn out like pandas. They are always chubby. They're not they have a lot of fur, and they have a lot of sometimes some animals have a lot of feather and stuff. This is because of their genes. This is because this is an adaptive feature that will help them to survive, because the fat layers actually act as insulation, and this insulation will prevent them from getting cold, and so that they can live in their environment, and also the fur may keep them warm as well. Extended students explain the adaptive features of hydrophytes and xerophytes to their environments. Hydrophytes are those plants that live in the water, such as the water lilies. There is adaptive features for them. You can see them in your book. Now, 18.3 selection. Describe natural selection with reference to genetic variation within populations. Production of many offspring. Struggle for survival, including competition for resources. A greater chance of reproduction by individuals that are better adapted to the environment than others. These individuals pass on their alleles to the next generation. So this is the process of natural selection. You have to know about it. Number 2 says, describe selective breeding with reference to selection by humans of individuals with desirable features. So selective breeding is actually done by humans, like farmers. Sometimes, for example, if they want only for example, let's say black cows. They only want black cows, for example, because they produce a specific amount of milk. So they will get the black cows and make them breed together, so that they will produce a population of the black cows only, and the other ones that they do not like, they will not let them reproduce. So they will only let the ones that with desirable features, the stuff that they want, will reproduce until they create a population of them, and they will only select them to survive and produce. So crossing these individuals to produce the next generation, selection of the offspring showing the desirable features. Number 3, outline how selective breeding by artificial selection is carried out over many generations to improve crop plants and domesticated animals, and apply this to given contexts. Now, extended students, they have to describe adaptation as the process resulting from natural selection by which populations become more suited to their environment over many generations. This is a definition. Also, this one is a definition. Describe the development of strains of antibiotic-resistant bacteria as an example of natural selection. So sometimes the antibiotics, as I explained in another chapter, in the drugs chapter, they are actually may grow resistant, and then it will be harder for the human or the person with the sickness to treat themselves, because they have the antibiotic resistance. So it's very hard in these situations, and these people are they didn't follow the protocol when they are using antibiotics, such as letting yourself take the antibiotic. Even if you feel better, just follow off the course and finish it till the end, so that there is no antibiotic resistance. Now, number 6, outline the differences between natural and artificial selection. 19, organisms and their environment. This is chapter 19 out of 21. There's only 3 chapters left, and this chapter, I think it's overlooked by a lot of students. I don't know why, but you have to focus on all chapters. Just because the chapter is at the end, this doesn't mean that you have to overlook this chapter, because it comes in a lot of exams at the end. So you have to make sure to focus and study on all chapters, not just the chapters that you think are important. You have to study everything equally. 19.1 Energy flow. Core and extended students state that the sun is the principal source of energy input to biological systems. So the sun will always start the energy like charts and the energy flows in all in all food chains. Describe the flow of energy through living organisms, including light energy from the sun and chemical energy in organisms and its eventual transfer to the environment. 19.2 Food chains and food webs. Describe a food chain as showing the transfer of energy from one organism to the next, beginning with a producer. So producers are always plants, like 100%. They are always plants because the plants, these are the old organisms that produce energy from their own. They don't need like something to eat so that they can produce energy. This is by photosynthesis, which I've explained in chapter of the plants. Number 2 says construct and interrupt simple food chains. So sometimes you will be asked to make your own food chain. You always started with the producer, and then you follow it with the primary consumer, and then the secondary consumer, tertiary consumer. So this is like the sequence. Now, these are a lot of definitions. From here until number 9, these are definitions. Describe a food web as a network of interconnected food chains and interrupt food webs. Describe a producer as an organism that makes its own organic nutrients, usually using energy from sunlight through photosynthesis. So as I said, it's always a plant. Describe a consumer as an organism that gets its energy by feeding on other organisms, such as humans or animals. Number 6 says, state that consumers may be classified as primary, secondary, tertiary, quaternary, according to their position in a food chain. So primary is the first one, secondary is the second one, tertiary is the third, quaternary is the fourth one. Number 7 says, describe a herbivore as an animal that gets its energy by eating plants. So herbivores only eat plants. They are vegetarian. Describe a carnivore as an animal that gets its energy by eating other animals. So they eat meat. Describe a decomposer as an organism that gets its energy from dead or waste organic material. So decomposers are the bacteria or organisms that actually feed on like waste organic material or like dead bodies. Use food chains and food webs to describe the impact humans have through over-harvesting of food species and through introducing foreign species to a habitat. Draw, describe, and interrupt pyramids of numbers and pyramids of biomass. Number 12 says, discuss the advantages of using a pyramid of biomass rather than a pyramid of numbers to represent a food chain. So pyramid of numbers is not accurate as much as the pyramid of biomass because sometimes there is high numbers of producer and then there is high numbers of there's not a lot of consumers, so it's not really accurate. It will not show amount of accuracy like a biomass. Describe a trophic level as the position of which an organism in a food chain, food web, or ecological pyramid. Now for extended students, draw, describe, and interrupt pyramids of energy. 16 says, discuss the advantages of using a pyramid of energy rather than the pyramids of numbers or biomass to present a food chain. The rest of it is over here. 19.2 continued. Number 14, identify the following as trophic levels in food webs, food chains, and ecological pyramids: producers, primary consumers, secondary consumers, tertiary and quaternary consumers, as I explained in previous points. Extended students, explain why the transfer of energy from one trophic level to another is often not efficient. Explain in terms of energy loss why food chains usually have fewer than 5 trophic levels. So sometimes energy is lost between trophic levels, not because the animal ate it, but sometimes they are lost, for example, in animal waste, or they are lost to the environment. So not 100% of the energy is always used. There is always some waste energy. That's why there's always fewer than 5 trophic levels. They are only 4 maximum consumers because there's not enough energy to sustain the rest of the food chain. Explain why it gets more energy efficient for humans to eat crop plants than to eat livestock that have been fed on crop plants. So It is always better for humans to eat, focus their nutrition on plants because they always have higher amounts of energy than meat or any livestock animals. 19.3 nutrient cycles describe the carbon cycle limited to photosynthesis, respiration, feeding, decomposition, formation of fossil fuels, and combustion. Now, the carbon cycle, if you've already taken chemistry, you will know more about it than here. So, this is basically like an ecology part. Extended students describe the nitrogen cycle with reference to decomposition of plant and animal protein to ammonium ions, nitrification, nitrogen fixation by lightning and bacteria, absorption of nitrate ions by plants, production of amino acids and proteins, feeding and digestion of proteins, deamination, denitrification. So this is the whole nitrogen cycle over here. Number 3 says state the roles of microorganisms in the nitrogen cycle limited to decomposition, nitrification, nitrogen fixation, and denitrification. The generic names are not required. Now 19.4 is about populations. Core and extended students have to define all of this. Population as a group of organisms of one species living in the same area at the same time. Community is as all the populations of different species in an ecosystem. Describe an ecosystem as the unit containing the community of organisms and their environment interacting together. All of these are, not all of these, it's from 1 until 3. These are very important definitions. Number 4 says identify and state the factors affecting the rate of population growth for a population of an organism limited to food supply, competition, predation, and disease. Sometimes the rate of population, their growth will not be as required because sometimes there is a shortage in food. There is competition over food, so that not all animals or organisms will eat. Predation, sometimes there will be attacks on them, and diseases may actually reduce their population. Identify the lag, exponential log, stationary, and death phases in the sigmoid curve of a population growth for a population growing in an environment with limited resources. Interrupt graphs and diagrams of population growth, so you have to know how to understand them, and sometimes they will ask you about identify the stationary phase, identify the death phase, so you have to know when and what happens during each phase. Number 7, explain the factors that lead to each phase in the sigmoid curve of the population growth, making the reference where appropriate to the role of limiting factors. So you have to, as I said, you have to know each phase and how it happens and why it happens, and you have to also label. Chapter number 20 is about human influences on the ecosystems. Core and extended students have to describe how humans have increased food production limited to agriculture machinery to use larger areas of land and improve efficiency. So humans actually increased food production, um, so that it can meet the increasing population needs. Chemical fertilizers to improve yields, insecticides to improve quality and yield. Insecticides are chemicals that kill insects. Herbicides to reduce competition with weeds. So herbicides actually kill weeds. Weeds are like the little green plants that come out. These actually take away the nutrients that the actual fruits and vegetables are needed to grow. So these are usually killed by herbicides. Selective breeding to improve production by crop plants and livestock. So selective breeding also applies to plants. Like I said, some farmers will only allow the animals with desirable features to grow. The same thing can happen in the plants. Describe the advantages and disadvantages of large-scale monoculture of crop plants. So monoculture is growing like a whole farm of the same type of plants. So for example, they grow a whole farm only strawberries. They don't grow anything else. So this is monoculture. This is it can be a good and bad thing. A good thing because it may increase the yield production for the increasing demand. For example, if some people demand this, they will get increased stocks of that. But it can be a bad thing. So if they all have the same kind of genes, of course, because these are they are all the same types of fruits. So if there's a disease or any predation, it will kill them off, and the farmer will lose like all of the yield and all of the crops, so it will be a big loss. Now, 3 says describe the advantages and disadvantages of intensive livestock production. Intensive livestock production is, for example, growing a lot of animals in the same crowded place. So the disadvantages of this is because it's animal abuse. Like if you get a lot of chickens, for example, in like a one farm, like it's going to be a lot, and it's also going to decrease like their like quality, and it's going to be not healthy. Like if they leave it out in the farm, they leave the animals to like run in the farm or something, it's going to be better, of course, than just crowding up a lot of animals, and it's also going to produce a lot more pollution. Advantages are there's a lot, of course, of chickens that are produced so that it can meet the requirements of the increasing population. 20.2 Habitat destruction. Describe biodiversity as the number of different species that live in an area. This is a definition. Describe the reasons for habitat destruction, including increased area for housing, crop plant production, and livestock production. So, habitat destruction is, for example, cutting off trees, and these trees may have some organisms living on it, so they are destroying their homes. Just imagine living in an area and like suddenly they come out and they just destroy your whole house and the whole area where you live. So this is habitat destruction. So they may do that so they can increase area for housing, crop plant production, and livestock production. So they may cut out like areas where there is lakes and there's trees so that they can increase the area where there is houses so that they can increase housing for the increasing population. And they may do that so that there is more areas and space for farms that can produce plants and livestock production. Extraction of natural resources. So for example, if there's a tree or a rainforest that they discover there is some minerals or some stones or something that is very precious beneath it, they will cut out all of the trees so that they can extract those minerals. Freshwater and marine pollution. Um, so they may also destroy the habitat for freshwater and marine pollution. State that through altering food webs and food chains, humans can have a negative impact on habitats. So if they cut off trees, they will actually destroy the producers, and then there will be food chains that are destroyed forever because they don't have the producer or they don't have consumers. Explain the undesirable effects of deforestation as an example of habitat destruction to include reducing biodiversity, extinction, loss of soil, flooding, and increase of carbon dioxide in the atmosphere. So, if you cut off trees, then you will have reduction on the biodiversity. All of the genes will be nearly the same. So, if there's a disease, it will cut all of the it will destroy all of the population right away of like a crop or an animal because there's no biodiversity. Extinction, some animals may go extinct because they can't find a place to live. Loss of soil, because there's no trees, of course. Flooding may occur because if the rainforest actually protects the place from flooding. An increase in carbon dioxide in the atmosphere. This is because there is no plants or trees to take in the carbon dioxide and perform photosynthesis, so there is higher amounts of pollution in the air, and greenhouse gases will increase and it will cause global warming. So this is a big problem actually. 20.3 Pollution. So core and extended students have to describe the effects of untreated sewage and excess fertilizer on aquatic ecosystems. So untreated sewage is very unhealthy for both plants and us humans because untreated sewage can actually make cholera to break out, and a lot of bacteria and life-threatening diseases can spread through untreated sewage, which is contaminated water, and excess fertilizer can cause eutrophication, which I'm going to be explaining now. Eutrophication is only for extended students, so if you're a core student, then you don't have to know about it. Number 2 says, describe the effects of non-biodegradable plastics in both aquatic and terrestrial ecosystems. Non-biodegradable plastics can actually cause a decrease in the fish population because the fish can accidentally swallow the non-biodegradable plastics, and then their stomachs will be full and they won't be able to eat anything and they may die. And also terrestrial ecosystems like the human ecosystems, these are also not healthy for us. Describe the sources and effects of pollution of the air by methane and carbon dioxide limited to enhanced greenhouse effect and climate change. So, as I said, if there is higher carbon dioxide in the air, then there is enhanced greenhouse effect. There is higher global warming. The atmosphere will always be hot, and ice glaciers may melt down. So, this is a big problem. And when ice glaciers melt down, there will be flooding and there will be tsunamis, so this is very, very dangerous. Extended students explain the process of eutrophication of water limited to increased availability of nitrate and and other ions. So this is caused by excess fertilizer in the water. So a factory may drop or may throw away a lot of fertilizer into the lakes. So this will cause nitrate to be excessively available in the water, and this will cause increased growth of producers. So there will be a lot of algae growing in the water, and this will take away the nutrients and oxygen from the water because as we all know, it requires respiration to grow, so it will take the oxygen away from the water, causing increased decomposition after death of producers. So a lot of other aquatic organisms will die. So this will cause the fish to not find food, and so increase the aerobic respiration by decomposers because a lot of the animals will die basically, and the plants also, their food sources will die. So there will be a lot of decomposers in the water that will actually work by aerobic respiration, and it will lead to further reduction in the dissolved oxygen in the water and death of organism requiring oxygen in the water. So this is all caused by excess fertilizer in the water. You have to, of course, know about this process and memorize it because sometimes a 6 mark question will come in the exam and it will ask you to describe the eutrophication process. So this is very important, and don't overlook those chapters because they are important. 20.4 is conservation. Core and extended students have to define sustainable resource as one which is produced as rapidly as it's removed from the environment so that it does not run out. For example, oxygen. Oxygen is a sustainable resource, it will never run out. And state that some resources can be conserved and managed sustainably, limited to forests and fish stocks. So you can protect forests and fish stocks by preventing them to be fish. Like for example, if there's some extinct fish that is going to be extinct, you will by law like protect them by not fishing them. Or forests, they will be not allowed to be cut down. If they are cut down, there will be serious consequences to the person who does this. So explain why organisms become endangered or extinct, including climate change, which is, for example, if the environment is getting too hot, some animals will not be able to live because they are used to cold weather. Habitat destruction, for example, some trees are cut down, so birds will go extinct because they can't find a place to live. Hunting, over harvesting, and pollution and introduced species. Describe how endangered species can be conserved, limited to monitoring and protecting the species and habitats. This is by like creating laws and introducing fines so that the people who do hunt down those animals, they will be paid a lot of fines, and there will be consequences to doing that. By law, education, by educating students and people and providing awareness to people that some animals will go extinct, there will be education and there will be less hunting down. People will understand the consequences. So basically, what we're doing right now is I'm giving all of you awareness about these problems. So this is a form of awareness given to people by education. Captive breeding programs, by for example having a panda. For example, some actually zoos, they do this. They will get a panda and they will force their like fertilization by maybe by maybe IVF. If you took it in the previous syllabus, IVF is like taking the semen from the male panda and like fertilizing it with the female panda. So this is captive breeding. They will actually breed together forcefully so that they don't go extinct. Seed banks like keep seeds of several plants that are going extinct. You will store them under the right conditions so that they don't germinate. Uh, Supplement students, extended students, explain how forests can be conserved using education, protected areas, quotas, and replanting. So, what is quotas? Quotas is usually done for fish. So, for example, there will be a specific time where people are going fishing, and there's seasons for those. Sometimes the fish will have seasons where they breed, and then the governments will prevent fishing during that season of breeding so that they don't go extinct and they don't fish out baby fishes. And this is to prevent the over hunting of or over fishing of some fish. And replanting is taking the seeds from the seed banks and planting them so that they don't go extinct. Explain how fish stocks can be conserved using education, closed seasons, as I just said, protected areas, controlled net types and mesh size, quotas, and monitoring. Describe the reasons for conservation programs limited to maintaining or increasing biodiversity, reducing extinction, protecting vulnerable ecosystems, maintaining ecosystem functions limited to nutrient cycling, and resource provision, including food, drugs, fuel, and genes. So sometimes we need to keep stuff from being extinct because they are being used for very important stuff, such as food, or sometimes they're used in medicines so that people can treat their diseases, and sometimes they're used as fuel, and they are used for genes so that there is increased biodiversity. Number 8 says describe the use of artificial insemination and in vitro fertilization, which I just described in the captive breeding programs. So I've explained that already. If you're a core student, you don't have to know about it. You can just listen to what I said so that you can understand more. You have a deeper understanding, and so you can really understand what's captive breeding. Explain the risks to a species if its population size decreases, reducing genetic variation. Knowledge of genetic drift is not required. So as I said, if there's risks if some population sizes decreases, like losing their like sometimes plants are important for drugs, so if they go extinct there will be a health problem spreading and people will die. So this is why some people or some organizations keep things from being extinct. 21 biotechnology and genetic modification. 21.1 biotechnology and genetic modification. Core and extended students state that bacteria are useful in biotechnology and genetic modification due to their rapid reproduction rate and their ability to make complex molecules. Um, Extended students discuss why bacteria are useful in biotechnology and genetic modifications, limited to few ethical concerns over their manipulation and growth. So, in the past, they used to use animals and pets so that they can create genetic modification, and they use them in science experiments, and there was a lot of ethical problems, such as, for example, some people are against animal testing nowadays. So using bacteria is not going to like cause any ethical concerns because they are just bacteria. And the presence of plasmids, the bacteria are used for two reasons: because they have fewer problems, and they also have plasmids which is not found in some organisms. 21.2 biotechnology. Core and extended students describe the role of anaerobic respiration in yeast during the production of ethanol for biofuels. Describe the role of anaerobic respiration in yeast during bread making. Describe the use of pectinase in fruit juice production. So pectinase is an enzyme that is used to produce fruit juice. Investigate and describe the use of biological washing powders that contain enzymes. So sometimes detergents and things, they will have biological washing powders so that they can remove lipids. For example, they can remove proteins or some stuff like that. Extended students explain the use of lactase to produce lactose-free milk. So lactase can be used to break down lactose in some dairy products so that people who are allergic can still eat dairy. Describe how fermenters can be used for the large-scale production of useful products by bacteria and fungi, including insulin, penicillin, and mycoprotein. Describe and explain the conditions that need to be controlled in a fermenter, including temperature, pH, oxygen, nutrient supply, and waste products. So fermenter is basically for producing large scale production of useful products by bacteria and fungi, such as maybe fermenters can be used to produce ethanol, which can be used as a biofuel. And also there is specific conditions like the enzymes. They have to have temperature, optimum temperature, and oxygen. And they also have to basically, if the oxygen levels are going low, then they always have to change it out. And the waste products have to be removed periodically, so they don't just leave it around. They have to always look and observe those fermenters so that the batch does not go bad. 21.3 Genetic modification. Describe genetic modification as changing the genetic material of an organism by removing, changing, or inserting individual genes. This is all that's required for students of core. And now for extended students, they have to know a lot more information. Outline the process of genetic modification using bacterial production of human protein as an example, limited to isolation of the DNA making up a human gene using restriction enzymes forming sticky ends. So genetic modification can they do it on bacteria so that they can produce human proteins. First of all, they will cut the area of DNA using restriction enzyme. Cutting of the bacterial plasmid DNA using the same restriction enzymes, forming complementary sticky ends. So they will cut out DNA from both the human and the bacterial plasmid and inserting the human DNA into bacterial plasmid using DNA ligase to form a recombinant plasmid. So recombinant plasmid is like having both the human and the bacterial DNA so that they can produce the protein. Insertion of recombinant plasmids into bacteria and multiplication of bacteria containing recombinant plasmids, expression in bacteria of human gene to make the human protein. So this is a process you have to know about, and you also have to memorize all of this. 21.3 Genetic modification continued. So we have to know some examples of genetic modification. So like the one I just explained, the insertion of human genes into bacteria to produce human proteins. There's also another one, the insertion of genes into crop plants to confer resistance to herbicides. And this is another way that farmers can actually take the plants and genetically modify them and let them reproduce so that they can protect the plants instead of using herbicides and pesticides, which actually can cause eutrophication and problems with the environment. So this is a way they can do resistance for the plants. The insertion of genes into crop plants to confer resistance to insect pests, the insertion of genes into crop plants to improve nutritional qualities. So sometimes they will genetically modify, for example, lettuce so that it can be more nutritious than the actual natural lettuce. Extended students discuss the advantages and disadvantages of genetically modifying crops, including soya, maize, and rice. So sometimes when you go to the supermarket and buy some food, it will be written on it sometimes non-GMO, and sometimes they would write contains GMO ingredients. So what does that mean? GMO means genetically modified food. So ingredients. So some people may not prefer that. Some people only want natural organic food, and GMO is technically not organic, so people may not eat that and they may not buy that. Some people are okay with it. And sometimes companies do GMO ingredients so that they can maximize their profits. And sometimes they are harmful for people, and they still do it because they will get more profits that way. They can have a large yield of plants. So some people actually may not prefer that, but it can be better for the company to produce GMO than not GMO, because it will be cheaper for both the consumer and the company to produce GMO things, and some they are not natural. So in the end of the day, it depends on who is buying the food and what they prefer. So actually, this is the end of the syllabus. You do not need to know anything else. This is all that's needed for both core and extended students who are studying for Cambridge IGCSE Biology. So I guarantee you that if you understand everything that's written in the syllabus and you memorize all the definitions, then you will get a 9 or an A star. You will get the highest mark possible. And thank you all for watching.